Hello, everyone, and welcome to VIMS, Virginia Institute of Marine Science, and to the Virginia Harmful Algal Bloom Task Force meeting. Glad you all could make it, and there should be probably a few more people joining us as we go on. If you didn't see, I'm sure you did, but there's plenty of food out there. As Savannah, my student, said, we had a muffin bloom this morning. They gave us a little bit extra food, more than we expected. So please help yourself, and please feel free to go out and help yourself to more coffee, tea, yogurts and muffins and pastries as the morning goes on. If you haven't already, just a few housekeeping items. The restrooms are out here down the hall to your left. When we break for lunch, we're going to break for lunch and go to classrooms A and B and C. It might be warm enough by then that some of you want to eat outside. There are some picnic tables behind Chesapeake Bay Hall. There are also some out in the area out this direction. So if you want to eat outside, you might want to eat outside when it comes that time. But the food will be located hopefully at the back part of classrooms A and B, and that'll be down the hall and to your left. So if you go out here to your right and down the hall to your left, that's where you'll find food when it's time to break for lunch. And we have a lot of food for lunch, so please feel free to come back for seconds and help yourself to a lot, because they did give us a lot of food when we went to pick it up this morning. So feel free to take some and take some extra if you want to take some home as well. We have lots of really interesting talks this morning and this afternoon. So this morning there'll be some monitoring talks, and then we have several lightning talks from students and postdocs. And this afternoon there'll be some updates from various labs on their research progress. And Danielle Schools will be welcoming us from the Virginia Department of Health.
Thanks, Kim. I'm going to try to talk without this microphone. Can y'all hear me? Okay. Yeah. Can you all online hear me?
They will answer.
They will answer their mute. Okay.
Oh, they can text them.
Okay. Good morning.
Good morning.
That is so pitiful. I know it's Monday morning. Let's restart. Good morning.
Good morning.
All right. At least we're awake. We got coffee, we got sweets, we got fruit, we got all kind of sugar stuff to keep everybody going. So my name is Danielle Schools. I am the director for the Division of Shellfish Safety and Waterborne Hazards. I work in the Office of Environmental Health Services with the Virginia Department of Health. We have some of our staff here today. Those of you who work at VDH, raise your hand. Yeah, so we've got a lot of VDH people along with some of our other partners for this. I just want to welcome everybody. Thanks everybody for coming down on Monday morning, as well as for those that are joining us virtually. This is some exciting stuff. We get to come together once a year for the annual HAB task force meeting to discuss all these things and share information and share data. Really excited about the student presentations. It's a great opportunity for you all to present, as well as for us to hear the work that you're doing, because that data is integral to what we're doing on a day-to-day basis. I just want to take a second and just recognize the work that all of the partners do with this HAB program. HABs, with the world the way it is today with climate, it's not anything that's going to be solved as far as the problems that HABs present. Whether it's recreational impacts, it's economic impacts, as well as for, at least from my side of VDH, we're really concerned about the impacts and possibilities on shellfish industry, as well as public health implications. I think we've seen over the last couple of years, HABs become more and more of a prominent public health concern as we've had more activity in different water bodies, both inland and marine. I also want to just take a second to recognize the work that everyone does. We had a leadership meeting with my group on Friday, and we talked about a team of teams. And so the work that's done in the state of Virginia really is work done because of a team of teams. We've got partners from DEQ, VIMS, ODU. We're bringing on new partners from other universities, as well as participation from some civic organizations and those that are actually being directly impacted by these harmful algal blooms that occur. I think the challenges that we face are continually trying to keep up with how these organisms function and how these HABs impact stuff, as well as limited resources. We are constantly pressed to do more with less, and we are constantly pressed from the public's needs at what is going on, why is this going on, how can you fix it? So it's a concerted effort that we're still pushing through, meeting some of those challenges, again, with limited resources. And I think that's a testament to the work that we have been done because of the people that are involved. I think one of the things that in my tenure with the health department for 25 years, especially with my staff now, I'm always cognizant of the fact that the research that's done, the work that's done, the response that's done, a lot of times is successful because of the people and not because of the resources. I think that's one thing from a government entity that we always struggle with, is that a lot of times our resources are very limited. So I just want to take a second to just recognize and applaud those efforts of everybody who's a part of that team and contributes to these HAB responses and studies, and just the general, just how we approach and communicating to the public, which is a huge thing. With public health, obviously, besides issuing advisors and whatnot, we're also trying to do education. I know my staff, specifically Margaret, Todd, and even our marine scientist folks, and Lindy, I spend a lot of time talking about HABs with people, answering questions. And it's not always public health as far as the human aspect, right? It's also, you know, last couple of years, we've had the things about folks, four-legged babies drinking out of retention ponds and whatnot. So now it's really gotten expanded beyond what it was several years ago. So I just want to welcome you all, applaud all the efforts that everybody has done for the past year and for the work that is to come. And with that, I'll turn it over to Margaret.
Thank you, Danielle. All right, our first lightning speaker of the day is Dante Hormans from VIMS. His talk is real-time forecasts of harmful algal blooms using empirical habitat suitability models. And I'm going to just read part of his bio. He said before he started as a postdoc research associate at VIMS, he obtained a bachelor's and master's degree in theoretical physics, did PhD research at the Department of Biology in Antwerp, Belgium, in close collaboration with the Institute of Applied Mathematics. He has been studying recently at VIMS, working on effects in environmental conditions on harmful algal blooms. So thank you.
Yeah, that's correct. Thank you for the introduction. And welcome, everyone. Good morning. The first talk, the lightning talk, is on modeling. So I think that's a great start of our day. So I will talk about how we can use models to forecast harmful algal blooms. And before I start, I would like to introduce you to the tool that we have. Most of you will know this tool. It's called Chesapeake Bay Environmental Forecast System, or CBEFS in short. And this tool, it's like a weather forecast, but we forecast the environmental conditions in the Chesapeake Bay. And on this slide, I show you the front page of the website that we have, where we publish 2-day forecasts of the environmental conditions. On the left, you have the list of environmental conditions, such as physical environmental conditions, temperature, salinity, and other factors, but also biogeochemical ones, like pH and so on. And what we do is each day we publish these 2-day forecasts, and we publish maps, spatial maps, also seasonal trends, and we compare with data and also depth profiles. How do we generate these forecasts? It's based on a mechanistic model, a very brief introduction. It's a 3-dimensional hydrodynamical biogeochemical model, or it's called ROMS-ECB. So it resolves the tides and also the biogeochemistry. It's based on the regional ocean modeling system, ROMS in short. It has a 600 meter by 600 meter grid and 20 vertical levels. And if you want to know more about it, Beaver et al. 2021 and Saint-Laurent et al. 2020. But we are interested in harmful algal blooms. And so the idea is to extend this tool, this forecast, with forecasts of harmful algal blooms. And how can we do that? we will use these 2-day forecasts of temperature, salinity, pH, so the environmental conditions. And based on the forecast of the environmental conditions, we will forecast harmful algal blooms. And this using a statistical model. And in the end, we want to map, as shown on the right, where you see where we expect ideal conditions for blooms, where we expect poor conditions for a bloom. But that's the idea, that's the objective. And to construct such a model, there are definitely some challenges. And today I will talk about one question that we had. We applied a statistical forecasting model using mechanistic model output provided by CBES, these 2-day forecasts of temperature, salinity, and the other factors. And we asked ourselves, should we train this statistical model using mechanistic model output, or can we just train it using in situ observations? And why is this question important? Because traditionally, we train statistical models using in situ observations. And you can, if you think about it, we know that, of course, mechanistic model output is not perfect. So the error in these 2-day forecasts may propagate as an error in the statistical model. That's basically the idea. That's what we wanted to study. And to answer this question, we came up with the following model experiments. So we tried 3 methods. In the first method, we will train a statistical model using in situ observations, and we also apply the model using in situ observations. And we call this method OO, observations. In the second model experiment, we train again the statistical model using observations, but this time we apply it using the mechanistic model output provided by CBES. And then in a final model experiment, we train the model using mechanistic model output. So we generate new statistical models, and then we also apply them using mechanistic model output. And then finally, we compute the model skill and we compare the model skill to determine which method is best. Okay, methodology, very briefly, we need observations, we need data, training data, and we need a statistical model. Let's start with the in situ observations. So here we used the Chesapeake Bay program data. We focused on 9 environmental variables that we know affect blooms. And we also focus on 7 algal taxa, and they're listed here. Most of them are harmful except Heterocapsa. And as you may know, Chesapeake Bay program, they publish cell count data. But to simplify the problem, we transformed these cell counts to binary bloom, no bloom data. And we did that using fixed thresholds, cell count thresholds for each taxon based on the literature. Okay, the mechanistic model output. We also need mechanistic model output to train and apply the model. So here we just use the output generated by CBES, by our forecasting tool. And finally, we need a statistical model. Here we used a generalized linear model. Brief introduction for those who don't know it. It's similar to linear regression, but instead of looking for a line that goes through your data, you look for this sigmoid curve. And in our case, we have bloom, no bloom data, so binary data. And so depending on an environmental condition, you just look for this sigmoid curve. And once you know this sigmoid curve, once you have trained the statistical model, you can compute the probability of bloom occurrence, as shown on the y-axis, based on an environmental condition, in this example, water temperature. Okay, let's have a look at the results that's shown here. So on the y-axis, I show the accuracy, and on the x-axis, I show the 7 algal taxa we focus on. And then I'll show the results, of course, for the 3 methods that we used. So let's have a look at the first method, the method in which we trained and applied the model using in situ observations. So this is depicted by the black dots here. And what we see is that we get accuracies, typical accuracies of high 70s, 80%, which is pretty good given the simplicity of our model, the generalized linear model. But what's interesting is when we look at the methods, the second method, so method observation mechanistic model, OM, so depicted in blue. So here we trained the model using in situ observations, but this time we applied it using mechanistic model output. And what's interesting is that you see this drop in accuracy, especially for these taxa here. And the drop can be pretty significant, more than 10%. What's even more interesting is that when we train a statistical model using mechanistic model output and apply it using mechanistic model output, this is depicted by the triangles, we can correct for this mismatch in the mechanistic model output. So we see an increase, overall increase of the accuracy, and this is depicted by the triangles. So this means that the take-home message is statistical models trained using in situ observations are less accurate when we apply them to model output, and then we apply them using in situ observations. But we can enhance the model prediction skill just by retraining the statistical model using mechanistic model output instead of in situ observations. And this is an important insight because in the end, let's go back to our objective. We want to extend our CBEFS tool with forecasts of harmful algal blooms. And we can use these insights to improve our model to get more accurate models. And we started publishing our forecasts for percent and minimum on the website, and that's shown here. On the left, we have a nowcast for April 14, 2023. And then on the right, we have the forecast, 2-day forecast, April 16, 2023. And what you see is like these patterns that are well known of percent minimum in the upper region of the bay. And with that, I would like to thank you for your attention. And of course, I'm happy to answer questions. Thank you.
Thank you, Dante. We're going to hold questions until the end of our lightning
Oh,
talks.
yes. I didn't know that. Okay. All
We'll
right.
have some time right at the end. Thank you so much. So if you have questions, please hold them. We'll have chance at the end. Our next speaker is Josh Garber. He's with VIMS, and his talk is Insights into the Stability of 3 Phycotoxins in Response to Various Environmental Conditions. He's a third-year PhD student in Dr. Juliet Smith's lab in the Ecosystem Health section at VIMS. He's studying the distribution and fate of phycotoxins within the Chesapeake Bay. He received his undergraduate degree in chemistry and has been studying phytoplankton compounds since his undergraduate research. Thank you, Josh. Go ahead.
So today, can everyone hear me? First off? I'm also going to be giving a brief update on some of the stat stuff that I talked about last year and some other passive sampling stuff that I'm going to be doing. So to start off, I talked about this last year, so I'm not going to spend too much time on it, but essentially I want to see if I could shorten the construction method for spats down from 3 days to 2 hours, which I was able to do. And so our lab has completely moved over to the 2-hour construction time. If anyone has questions about this, feel free to see me after. I also briefly touched on this last year, but I have a bit more data for it. The objective of this was to determine how long you can store spats for once they've been produced and have them remain viable. So at the start of this experiment, I made a total of 30 spats and stored them in either 100% GenPure or 20% methanol for varying amounts of time. After those time points, which were 1 month, 2 months, 4 months, 6 months, and a year, I deployed them for a week alongside a control that was made the day before, so essentially no storage. And for all 3 treatments, I deployed them in triplicate. So brought them back to the lab and analyzed them for toxins. So on the y-axis, I have the concentration that I found in nanograms of toxin per gram fluorescent per day. And on the x-axis is the treatment, whether that was controlled GenPure or methanol. And I have found 5 toxins through the course of the experiment. For the first 2 months, I did not find any AS2, and for the first 3 sampling time points, I did not find any DTX1. The red boxes indicate that the amount of toxin that was absorbed was statistically lower than the control spats. I only compared the GenPure to the control and the methanol stored spats to the control, not between each other. And from this, looking at the 6-month data, both the spats stored in GenPure or methanol were statistically lower than the control spats that were deployed. And so from this, I concluded that spats could be stored up to 4 months in the refrigerator in GenPure water. And so that is what we have been doing for our lab since then. And so additionally, I wanted to look at a new type of passive sampler, as we know that spats aren't cannot be quantified and not the best for things like that. And so this new type of passive sampler is called composite integrative passive samplers. They were created by Amy and Shay, and they were created by impregnation of microfiber membranes with both hydrophilic and hydrophobic monomers. And these are different from passive samplers as they don't have that typical sandwich structure that most do, such as the mesh that's in spats with the resin in between. And so these passive samplers come infused with performance reference compounds to achieve for better quantification, which is an improvement upon the spat technology. And they are also customizable in terms of shape, size, and performance reference compounds that you can get infused in them. And so for some of these experiments, I plan to deploy both triplicate spats and SIPs off of here at VIMS. And I'm going to do this across a couple of seasons just to see how their performance behaves within different environmental moving on to some toxin degradation studies that I'll be doing. This is preliminary data from some experiments that I've done before. So on the graph, we have the time on the x-axis and the percent of the initial concentration on the y-axis. And for these experiments, I added toxins to artificial seawater in the dark with varying salinities, pH, and temperatures. These were held in quartz vials wrapped in foil in a water bath. And across the three preliminary experiments I did, the control conditions were a pH of 8, a salinity of 30, and a temperature of 35. So for example, in the salinity experiment in the middle, the pH for all those would have been 8 and the temperature would have been 35. The straightish lines across the top with the open diamonds are okadaic acid, and then the closed diamonds are azo-2. From this experiment, I found that the azo-2 half-lives range from approximately 7 to 16 hours, depending on what treatment it was in. And OA did not degrade, which is consistent with other papers that have been published thus far. For the three experiments, the azo-2 salinity of 30 treatment and the temperature of 15 degrees were statistically different from the other two treatments in their respective experiments. And so the reason that I've been interested in this and why I found this to be important is that from a study that Michelle Onofrio did from a previous lab member, she found that azo-2 was found pretty much year-round throughout multiple sites in the Bay. It was just not found within 2 of the sites that she did in her study. And so even though I found that azo-2 seems to degrade pretty rapidly, but we find it year-round in the environment, I'm interested in what's allowing for this and what factors are allowing it to persist more. And so the future directions that I will be doing, I will be repeating these experiments, but then also including yasotoxin and pentatoxin too. And then I also plan to do an experiment with lights and Hemix to investigate some direct or indirect lipolysis that may be happening. And I'm open to suggestions if anyone has them and wants to talk about this. And with that, I will thank everyone, my funding, and if anyone has questions, I will answer them later.
Okay, thank you, Josh. Our next speaker is Lex Berger with GMU, linking biological and functional microbial diversity with environmental conditions within the Chesapeake Bay watershed. Lex is a 4th-year PhD candidate in Dr. Jennifer Salerno's integrative microbiology ecology lab at George Mason University. Lex's research interests cover a broad range of microbial ecology topics, from bioluminescent symbiosis to bacterioplankton dynamics in estuaries. PhD work is focused on characterizing the biological and functional diversity of the microbial communities found in the Shenandoah and Potomac rivers across a range of environmental gradients. Bacterioplankton dynamics in both rivers have been severely understudied and have become a major concern in water quality. Thank you, Lex.
Before you would you mind using a microphone? People online hear better with the microphone.
Yeah. Is this better? Yeah. Okay. What? Good to go. Okay. Well, good morning, everyone. My name's Lex Berger. I'm from George Mason, and I will be talking to you today a little bit about my dissertation work. So a little background on kind of what I do. So as we typically see, trophic webs, food webs that we see in aquatic ecosystems, typically we see algae all the way up to, shorebirds, large fish, the whole typical stuff that we see. But really, this only gives us partial picture of what's going on in these ecosystems, right? All these interactions. So actually, bacteria do form the basis of our food webs. And, in our typical models, we don't see those. So I fixed it. So these are my guys. So why is this important for us? So we know bacteria are key players in nutrient cycling in, every ecosystem that we find, but especially in aquatic ecosystems. However, a lot of the work that we see, these bacteria are severely understudied, especially, in this region. So I'm hoping to answer some of that and provide baseline data. So George Mason was we were hired to work with the ICPRB and the USGS on their harmful algal bloom project in the Shenandoah River. And what's really cool is this can tie into the rest of my dissertation work. So as I've pointed over here, you know, we have the North Fork and the South Fork of the Shenandoah River. And up here is where the Shenandoah River meets the Potomac River. So my work will cover a little bit of both. So just wanted to point those two areas out real quickly. So the big question that I wanted to talk to you all today and kind of give you an idea of what work is going to be done in the next year is I'm looking at how does water quality and river discharge affect benthic cyanobacteria, spatiotemporal distribution and diversity in the Shenandoah River. So really what bacterial communities are there, especially benthic cyanobacteria? How do they follow the distribution over time and space? So we have, if I can do my math right, 10 sampling sites. We have 6 on the North Fork and 4 on the South Fork. And so the USGS has been collecting samples for the past year for us. And we will begin extracting soon. So kind of what we get is we get a bunch of these mats from different sites in the rivers. And we we call them a sausage roll. It's like really gross. But we kind of separate them in for a bunch of different analyses. So my work focuses on the molecular work. So on the right-hand side is about as much sample as we get to use. And with that, we'll go through extraction. So we extract the DNA from these guys, and then we're going to go sequence them. So a little background is, in case you don't have a lot of molecular familiarity. So we take our environmental samples, we extract the DNA out, we run a polymerase chain reaction, amplify that DNA, and then we're going to sequence it. So what's really cool about the work that we're going to be doing is we're going to be using nanopore sequencing, which is long reads. It gives us a long read and deeper reads. So we can actually have higher taxonomic resolution than typical molecular work that you see with like 16S rRNA. And so that resolution will be a lot better. So we can even get to the species level or strain level, which another lab in our program is actually working on the taxonomic, like the physical taxonomic work. So we're hoping to compare our molecular work with the physicality of that. And actually, I just was awarded a new project where we're going to work on hindcasting and forecasting the distribution of the cyanobacterial species based off of environmental conditions. So as we're using our molecular data, we will actually look for toxin-producing genes within these samples. And we're going to use those as a proxy for biomass in order to model how these cyanobacteria flow and work through the Shenandoah River. And we're hoping we can help predict algal blooms based off of this model, which is really cool. And so this kind of plays into our work also that I'm currently writing up in one of my chapters is and towards Alexandria in Hunting Creek and Gunston Cove, we've been doing some bacterioplankton work there as well. And currently we have some of our abundance data, our relative abundance data. And I just want to point out in this green, the lime green, and Synecocallis, this is where we find all of our cyanobacteria. And just really cool. We haven't seen any, we're hoping to see if we can see any similarities between Shenandoah and here with that flow through. And just, you know, as a higher resolution for that, just more of the species that we see. So we're hoping to work some more deeper analysis through that. And we're also hoping to work on hypoxic and anoxia models along with that Shenandoah model to see how all of that kind of interconnects with as all of these tributaries flow into the Chesapeake Bay. So yeah, so we're hoping to address some of these knowledge gaps, especially what toxin producing genes are there, distribution and species composition, working on these models and hoping to use this data to inform policy. I'd like to thank George Mason, the ICPRB, the USGS, the 4VA for funding us, and the ASLO LoRX program. And I'll take questions later. Thank you.
Thank you, Lex. Our fourth and final lightning speaker in our first session is Riley Hines with Ferrum College. Her talk is Preliminary Report on Correlations of Selected Cyanobacteria and Abiotic Variables in Smith Mountain Lake from 2022 to 2023. She is from Fauquier County, Virginia, currently a junior at Ferrum College, studying environmental science and agriculture, with an emphasis in crop science and a minor in biology. She's hoping to obtain a master's degree after undergrad and then plan on going into ecology or water conservation. Thank you, Riley.
Good morning. Also, a little bit about me. Last summer, I actually worked on the lake project, so I was firsthand doing all of the sampling. So I really have that understanding of the procedures for the analysis that I have worked on. And I've also been analyzing this data since I got to Ferrum as a freshman. So this is my third year doing it. And just a little bit of history about our project. This summer will be our 38th year, which is phenomenal. And we were co-founded by Dr. Carolyn Thomas and Dr. Dave Johnson. And this here is just a list of our sponsors and where we receive all of our funding. And so a little bit about our sample collection. We use an in situ aquatrol where we get all dissolved oxygen, temperature, and conductivity readings. And then in these two pictures, you will see our horizontal and vertical tow using a phytoplankton tow net, which this is just how we get the collection of our algae to bring back to the lab and count. And then we use a Secchi disc for Secchi depth. And this picture here is actually one of our volunteer monitors. We do not collect all of our data ourselves. We have monitors around the lake that go and they use what we call an integrated water sampler to really get a portion of the whole water column itself, where they take 50 mils and pour it back into a bottle for us that we use to look at total phosphorus using a latchet via the quick chem method. And so this is just a breakdown of what our summer looks like. It's 12 weeks, and in each period we see 2 weeks. And in the first week is where our volunteer monitors are out collecting, as well as the students. And we are looking at E. coli bacteria, but I really want to emphasize this is where all horizontal data is collected. And then week 2 is our depth profiles. And this is where we get all vertical data. And our volunteer monitors, they don't have to do anything for us this week. And then this here is a map of the lake. And I really want to point out right here, this is the Blackwater, really beginning over in here. And then we have the main basin and the Roanoke Channel. So you can really see we see higher levels in the upper parts of the two channels, and the main basin itself is pretty clear. And then this is just a map of all of the bacterial sites, which I said again, this is our horizontal data. This is our depth profile sites, which will be where all vertical data is collected from. So if we go back to the bacterial sites, it's going to be 14 sites, and depth profiles are 5 sites. And into my analysis, as a result of an atypical summer last year with the HABs, I wanted to really look at the abiotic factors that we already sample and then use that to see if I could find a correlation with the cyanobacteria counts. And I really want to emphasize this is not an experimental study, which is why you won't be seeing a hypothesis, but a purely observational study. And I use Pearson correlation coefficient. So all of my correlations, we're going to be looking at R and R squared, not R squared. So this map here is out of our report from the summer, which is what I took from our professors. So you're really going to see overall total diatoms. We saw more than twice as many in 2023 than 2022. And then total green algae, not quite as many, but I really want to emphasize that we don't count every species that's out there. We only count a select few of them. And then with our total cyanobacteria, so the first column, orange column, we're going to see this includes the 3 genera, which is the same that we counted in 2022. And you can still see we had twice as many. And then this hex column right here, I want to point out that we started counting 5 genera instead of 3 because we found Warrenichia and Aphanizomenon, which you can see was our primarily blooms we had over the summer. And then the star on this graph is just to emphasize, in period 2 is when we added those extra 2 genera, and that's when we began to see our bloom period, which really peaked in period 3, and then dropped back down here in period 4. So over total, I ran over 74 correlations, so you're only going to be seeing a few of them, and here I highlighted vertical temperature. This is one of our consistencies we have found within our data from 2022 to 2023, and we can see we have a strong correlation with temperature. And so this is just showing that the algae we're seeing, they prosper better in that colder water than they are once we're seeing the water warm up, which I found to be very interesting negative correlation. And it evens out a bit more when we're looking at the 5 genera. Over my data, I have really noticed that when we are looking at Warrenichia and Aphanizomenon, it really changes all of my correlations. And I believe that it may be because of their behaviors. So I want you to keep that in mind throughout the rest of my data. And here, once again, we'll be looking at vertical, but we're going to be looking at conductivity. So you're going to see 2 different boxes here. This first one is going to be conductivity over our whole lake. And down below the bigger box is going to be broken up by channel. When I was doing my data, I really wanted to see if I could find differences in the 3 separate parts of the lake. And I did, and you can see it across all of the data, but I really found it in conductivity. And in conductivity, I found it really interesting. It was extremely low in 2022, but when we look at 2023 with 3 genera, I have fairly strong correlations. And then once we add those other 2 genera we found over the summer, my correlations, they become weak to moderate and negative, which I found to be very interesting. There's not much literature on the behaviors of those 2, especially when you're looking at conductivity. So like I said before, I believe that we see this shift due to their behaviors, and that's something that we're hoping to possibly find more literature on or just begin to get a better understanding on them. And this has led us to wanting to possibly add more TP sites on our depth profile sites at the time, because when we go back and we look here, all of these sites that had our all of our high numbers in our outliers was one of our depth profile sites. So I have really been persistent with all of our professors at Ferrum on asking to add these sites so that I can get better data to look at. And we also want to take into consideration rainfall data with events, land use and water use, geographical features of the lake, because the 3 different areas of Smith Mountain Lake are very different. The Blackwater is more agriculture, the Roanoke is more industrialized, and then of course the main basin, that's where the dam is and everything else. And that's also the deepest part of the lake. And I also would really like to see if we could get an understanding of when we're seeing fertilizers being applied and coming into the lake, both commercial and private. And then all work I do is in honor and memory of Dr. Carolyn Thomas. I'm very thankful for her. I would not be here today if it was not for her. And I really just wish I would have gotten the chance to work under her. And then everything that I do is for who I call Mr. Murphy. He is from Shenandoah. He gave me my love for the water and really gave me my foundation for conservation. And I just, since 2019, his passing, have dedicated myself to proving that I can do this and that I can, as I used to tell him, save the water. And a huge thank you to all of my professors that have helped me. And questions or comments?
Good job, Riley. Thank you so much. We're gonna go ahead and accept questions first from the audience here. Are there any questions?
I have an online question. Go ahead. So it's a question from Shelley to Riley. Yes. She was wondering if this monitoring data is available.
Yes.
Yeah.
We have a website with all the reports. So the reports are there. If you want to.
So we publish our reports online, which is on our website, and you can go to the Smith Mountain Lake Association, where there's actually constant data there, that they do from that office, and part of it also is from what we do over the summers as students. So it is, you can go and see all the raw data as well.
Thank you.
A question for Dante. Why do you think you saw the drop in predicted results when you had inconsistent input and application data?
Why did I see the drop? So you're talking about when I train a model using mechanistic model output instead of in situ
Yes.
observations, right? Okay, so the only difference when you compare the methods is the in situ versus mechanistic model output. So the drop in accuracy is just caused by the fact that we do not model the reality perfectly using our CBEFS tool. So it means that an error in your mechanistic model output will propagate through your error as an error in your statistical model.
Right, but when you had an MM model, the accuracy was higher again. So
Yes.
does that mean it's a systematic error? So
yeah, it means that by retraining your model, you can partly correct for these errors.
Gotcha.
So that's the very nice and cool thing about our results.
Any other questions?
This is for the last talk. That was very interesting on Smith Mountain Lake. I think that the addition of maybe biovolume would be a great way to try to get around the size variation that you're dealing with. And may help explain some of the variance. This is a suggestion.
For years, we counted everything based and would use biovolume some, but with the cyanos being so intensive, we shifted to mostly cyano counting. But you're right. There's a big difference between a microcystis, and a chlamydomonas or one of the others. But yeah, I agree with you.
All right. Thank you so much for our lightning talk speakers and those great questions. We're going to move on to our next talk. We have Margie Mulholland from ODU, professor with the Department of Ocean, Earth, and Atmospheric Sciences. She's a professor in oceanography at ODU, biogeochemist, and leads the water column biogeochemistry group, as well as the phytoplankton analysis laboratory in the Department of Ocean and Earth Sciences. She has worked on harmful algal blooms for over 30 years.
Hey, everybody. Sorry, I cut it close here. We had everything bad that happened this morning could already. We got stuck at the tunnel on the first bit, and then the bridge was open and there was an accident on the bridge when we just came over. So I just walked in, so I'm a little frazzled, and my plane, I didn't get until 1 a.m. last night. So it's been quite the day, a morning already. But anyway, I'm going to give you a brief update of the estuarine HABs, and then this afternoon I'll talk to you about what our lab group is doing, and there'll be several different presentations by students that I'm really excited about, all the things we've been pulling together here in the lab. So without further ado, we'll talk about the 2023 estuarine HABs that we had this year. The samples we receive are from the Virginia Department of Health shellfish monitoring. There is bloom response as well that the Department of Health undertakes. And then we have several different monitoring programs that I'll talk about as well that we take advantage of. I'll talk about the notable blooms and then our expanded plans for the coming year because we've been awarded a HAB grant, which we're really excited about. Kim is a co-PI and Margie Friedrichs, I don't know if she's here today, but Dante's involved and quite a few people in the room are involved with that. Okay, so the shellfish monitoring samples, we get monthly samples from the different shellfish beds. We receive samples that are preserved with Lugol's iodine solution, and those come to our lab for phytoplankton analysis. We also get some unpreserved samples for toxin screening, and we use ELISA kits to do that. We had 510 samples this year. Sadly, we had some staff issues that happened this year. We had some health issues, so we got behind in the samples. So we don't have all of them analyzed for the bloom season this year, but hope to within the next month or two. We've analyzed about half of them. Then we get the bloom samples. Those are in response to bloom reports or visual observations in the field that field staff make, and then they'll collect a sample in response to that. We also have samples, as I said already, from VDH. We get samples, bloom response samples also from the Chesapeake Bay program. If they're out on the water and see discolored water, they'll grab a sample. And then the Hampton Roads Sanitation District, they run data flow cruises weekly, actually now bi-weekly, every 2 weeks in the lower James River. And when they see discolored water or when chlorophyll concentrations are above 25 micrograms per liter, they'll collect a sample for us and bring it to us. And then we have a time series site we've established in the Lafayette River. That's a subtributary of the lower James River. And we've been occupying that since 2012. And there we sample usually twice or 3 times a week during the summer. So we get a lot of samples from that place. And we get an increasing number of samples each year. The sample number has built based on observations and public awareness. So we're really excited about that. Okay, so this is a map showing where we get samples from. A lot of you may have seen this map a million times, so sorry. But in the blue dots, let's see, is that I got the laser, good. Okay, the blue dots are the Chesapeake Bay monitoring program stations. We have 7 rather, from the Chesapeake Bay and 7 from the tidal tributaries. Oops, that's not right. Yeah, that is right. So, and we get these from January to December. I've highlighted that because they stopped collecting samples for a while in the winter for us, but we've asked them to resume that sampling because there are wintertime blooms, but they're just under surveilled. And so when we had a bloom, it was in 2020, we realized we weren't getting samples, so we asked them to go ahead and keep collecting the winter samples again. So, and for the Chesapeake Bay program samples, we do full species composition. And yeah, sometimes we're missing samples because of weather when the ship doesn't go out. So that happens. The yellow dots, oops, wrong one. The yellow dots are the VDH shellfish sampling sites. And we get monthly samples from 69 states, and that's year round. And from these, we only do targeted HAB identification. Actually, this is going in and out. Can you guys hear me? Just loud? What's that?
People online.
Oh, I see. Okay. Thank you for that. Okay. So I'll keep using this, even though when I turn my head, I go out. Okay. So for the VDH samples, like I said, we get these monthly, but we don't do full taxonomies. We just do the targeted HAB species for these. And then there are some other CV tox samples. We only get a few samples from that. And then, like I said, we have a time series site and that is down here in the Lafayette River. And we sample this very frequently in the summer. We've actually just established a new time series site as well in the Elizabeth River. And that's related to another project, but we're excited because we're getting more and more surveillance down there. This area in the lower James is highlighted because in this area, that's where the Hampton Roads Sanitation District does their data flow monitoring. And they used to go out weekly, now they go out bi-weekly. But again, they pick samples up for us. Oops, I can do this. Okay, so this is just in words. We get the HRSD data flow samples every 2 weeks. We've been receiving the Bay program samples year round since 2021 now. And in essence, we started doing year round again because of the heterocapsa blooms in particular. Those were the ones we were missing in the winter. And the time series site, we have now gone year round on that. We used to only sample it in the summer. We're now doing weekly or bi-weekly or twice a week. The bi means both twice a week or once every 2 weeks. So, but we're doing twice a week now in that time series site as well. We added, like I said, the time series in the Elizabeth River, and we're doing continued work on our trying to get all these samples that we've been doing microscopically for years. We're now running them as well through flow cams and through planktoscopes to try to build our libraries, especially for the targeted HAB species that we've been looking at. So Kayla is going to give a little update on what we've been doing with imaging in just a little bit. But we started this work during the pandemic. And so I have Sophie's paper listed here about the different things that we were doing with the imaging during the pandemic. Okay, so now let's get to the blooms. And in general, this is a figure that was built off an old data set this is just 2007 through 2020 data, and essentially plotting the abundance of different taxa throughout the year. But essentially it shows the pattern that we get a very predictable seasonal progression of blooms in the lower bay, where we get heterocapsa species in the winter months, followed by P minimum in the spring, and then gymnodinium species, and in the summer, carlodinium, Akashiwo, Scripsiella, Margolophodinium, and Alexandrium in the summer. And they sort of progress as the summer goes on. Usually when we see Akashiwo, that's a trigger for us to go sample more often because Margolophodinium is not far behind. Okay, so this is just showing 2020, oops, gosh, sorry. This is just showing 2023 now here and 2022 for comparison, but this is our species abundance, and this is cells per milliliter. Sorry, that got cut off there. But essentially we had blooms of heterocapsa in the winter this year. And then this heterocapsa, and this is heterocapsa as well. And then we had some Margolophodinium coming up in the summer. But again, we don't have all of our counts done. So we know there was more Margolophodinium in the summer as well. So this was pretty much the same as in 2022, where we had as well the heterocapsa in the winter, and here we had Margolophodinium in the summer. So we didn't have a lot of some of the other species, but that happens in different years. Some years they are there, some years not. Okay, so the highlights here in words are that we had Heterocapsa rotundatum, as well as Triquetra between January and April, and then Margolophodinium in July and August of this year. And we observed some of the blooms at our time series station, but the Margolophodinium really didn't hang around at the time series station this summer. And we have some theories about that as well. And we have a lot of anecdotal as well as observational data suggesting they were in other places as well. For example, the March bloom was observed near Tangier Island. We just had trouble getting people out there to sample. And so this is something we need a lot of help with, and we're hoping with our upcoming HAB project that we'll have better responsive sampling to that. But again, more about that later. Kim, I know we'll talk about the Marge blooms later on. And now I'll show the maps that Todd made for me is of the different species that are of concern here in Virginia for Pseudonychia in 2023 rather. The figure on the left shows 2023 and the figure on the right shows 2007 to 2021 where the blooms happened. But again, the populations are happening all over in the lower bay. We're seeing Pseudonychia, okay, but they're generally in low cell densities, but we want to keep an eye on this one because it's a nasty one. So we're keeping an eye on it, but it's always less than 1,000 cells per milliliter there. Okay, and this is, so this is breaking down the Pseudonychia into the larger ones and the smaller ones. Again, they're all occurring at most of the stations that we're making observations from in the lower bay. But we haven't, again, had reports of big blooms of these. This is a figure showing Dinophysis. Again, these are generally at low cell densities, but they're widespread and throughout the Chesapeake Bay, as well as in the seaside along the eastern shore. And this is our 2023 counts over here, the ones that we have to date, as well as the whole time series over here on the right. But again, they're generally low concentrations. The higher end here is greater than 10 cells per milliliter for the biggest dots. So they're low in abundance, but this is a worrisome individual. And so we're keeping an eye on it. Okay, and this is Carlodinium, and I didn't have the time series here, but you can see again, these are generally at low cell densities, but they're everywhere. Oops. Okay, and here's Heterocapsa. And like I said, since 2021, we're again getting wintertime samples here. But in 2023, we were able to observe some blooms of these species, especially in the tributaries. And so we're keeping an eye out on these. And we had a paper come out in 2023, Nicole Way here at VIMS, headed that up. And just about the under surveillance of Heterocapsa blooms. And these are really important food web species for the Chesapeake Bay as well. Here is the map of the Porocentrum in 2023 here. Again, they were generally in 2023 at pretty low cell density, but they were everywhere. Again, so, and then this is again the sort of over the years view of them when they bloomed. And we didn't really see any pronounced blooms in 2023 of this organism. And we would have caught these ones because these samples are, oh, you did. Kim saying they did. So Kim will have a different story from further north. Okay. And then Akashiwo and Scripsiella, I put maps up for these guys. Again, no major blooms of these during 2023, but they were there. They're always there. And like I said, with the usual seasonality to the populations. And Margolefidinium, our favorite bloom former down where we are. And we don't have a lot of the samples that were analyzed yet for the summer, but we know there were blooms during 2023. They weren't as pronounced at our time series site, which is right down here, but we know they were very bloomy in the York River. And out in the main stem of the Chesapeake Bay, but we didn't observe as many in the lower Chesapeake Bay, and we think that has to do with summer temperatures getting warmer and us having heat waves in the summer now in the south. Okay, and I'm about done, so don't worry. And there's Alexandrium, and we just have a few data points for it for 2023 because we're still analyzing the samples, but we didn't have major populations of them in 2023. And finally, the program plans. I'm going to give my New Year's resolutions in the next talk this afternoon. So I'll just say how we did this year. We still have to catch up on our sample analysis. We're continuing with our merging of different databases for phytoplankton observation. We've resumed the HRSD data flow we did have a hiatus in those, and after the pandemic, they had forgotten to collect samples for us, but we reminded them, and so they're collecting samples for us. And we're continuing our sampling at the Lafayette River Time Series Station. We've added the one in the Elizabeth River that we're excited about. We've been building our FlowCam classifiers and working off our multiple programs and enhancing that with new partnerships as well. And we're doing year-round sampling and analyses. I've taken advantage of classes at ODU who are doing some of the sampling for us. And we're trying to, and I'm teaching a CURE class in the fall, which is a classroom-based undergraduate research experience. So we plan to use the time series and a lot of this harmful algal bloom sampling for that. And we have lots more undergraduate student involvement now in this program. So we're really excited about that. And I think this is all I had, is our New Year's resolutions will be coming this afternoon. I give a summary of what other things our lab group has going on. So thanks.
Thanks, Margie. We're gonna hold off on questions so we can catch up a little bit on time. Our next speaker is Todd Edgerton with VDH. He's gonna give the marine biotoxin recap for 2023. Todd has worked with VDH as the marine science supervisor for 8 years, and he is a member of the ISSC Biotoxin Committee and has worked with harmful algal blooms and phytoplankton monitoring in the Chesapeake Bay since 2001. Thanks, Todd.
For those that have seen this presentation.
Pick up your mic, please.
Two mics. Both work. Is this one working? Yeah. All right. Get oriented here. For those of you that have seen this presentation before, I've taken out a lot of the introductory material. Margie just did a great job with a lot of that. So we can kind of all focus right on the toxins this time. So we'll be talking about the marine biotoxins. This is our station at the James River. This is Hilton Beach. You can see we have shellfish deployment here. In the background, everything staged really well. We've got oyster harvest going on, some commercial activity. So again, a lot of you have seen this before. There's a lot of phytoplankton monitoring that's going on. But today, Margie's really talked about most of the cell count data already. And then Kim's going to be up next talking about the molecular work that leaves the toxins. So I'm going to be really focusing on the toxin work that we've been doing over the last several years. So we started putting oysters out in, I think, 2019 with Juliet's group. Prior to that, doing a lot of work with spats as well, going back to 2017. So now in 2023, we added another station in Chincoteague. We've had stations before here on the shore, and we were really seeing a lot of domoic acid from Pseudonychia, and we wanted to make sure we had an additional site. So we established this Chincoteague site up there in our northernmost growing area. We have to move these stations around a little bit. We've got now a station on the northern neck, and then we moved slightly our Rappahannock station. This is right here at VIMS on the VIMS Pier. The James, we've been using the James River Fishing Pier. Now we're upstream just a little bit at Hilton Beach. That's where that cover photo was. This is the Brock Center at Chesapeake Bay Foundation in Lynn Haven. The Wise Point boat ramp down here at the South Shore. And this is VIMS facility on Watch Creek as well. So all of these yellow sites we'll be doing biotoxin work from, and then these we have monthly collections of seawater as well as shellfish for the entire year this last year. So this, and some nice pictures of oysters. This is really here as a reminder for me to tell everyone just how much work this takes, and really thank all of the additional staff, not just our marine science staff, Jackie and Evan, they've gone out and really put out all of these oysters and collected all of these oysters, come back and done a lot of cleaning and shucking, but also all of the other field staff and lab specialists as well. We wouldn't be able to do this work if it wasn't for the entire team. So a lot of work goes into it. You can see here's our bag. We're using aquaculture bags. We're deploying on piers. We've got several dozen oysters out there at a time. All of the oysters are out there for 30 days at a minimum before we collect them to make sure that they have enough time to soak. And then we're harvesting these as well as collecting water samples. One thing that we did do, just quickly for those of you that are working a lot with oysters, we're using a slightly different shucker this year. I'm happy to talk to you a little bit more about it. It's a leverage, has a spike that gives us a lot more ability to, I'll definitely talk to you, Kim, because we've shucked hundreds of oysters this year, and this really it adds a lot. So it gives you that initial break here, and then you can go in and finish shucking it with the knife. So yeah, we can talk about that at lunch. It's a good lunch activity, talking about oyster shucking. It's a lot safer as well, instead of there's no one stabbing each other in the hand or anything. So these maps look very similar to what Margie just showed, but these are toxin maps. So she presented where the dinophysis was identified. I do want to point out that we had no concentrations over 10 cells per mil. That was the highest on there, but we didn't have any of that from those last maps. But here, this is toxin concentrations. This is everywhere we've done the okadaic acid ELISA. So these white dots, we ran the analyses and they're all below the detection limits. And we had 3, though, where there was detectable okadaic acid. And then this is a time series of dinophysis counts, so cell counts here. And then if they're surrounded by green, that means that they were okadaic acid negative, and our 3 positives are down here. So once again, the story that we've heard before is that we're not seeing a great match between cell counts and toxin concentrations. So here's all of these cells, up to over 2 cells per mil, which is, again, relatively high for dinophysis. This is a really low species, low concentration bloom former, if that's the term. But the 3 that we had positives for were in samples where we did not detect dinophysis. So it's a frustrating scenario using this as a tool. It's not a great tool to measure for dinophysis. So that's why we're really continuing to make sure we have ways of measuring the toxin directly. But the 3 places where we did see okadaic acid here, the James River and Lynn Haven, right in the second half of the year here from August to November. I don't have toxin data for shellfish to show today. We're still working on revising that method, trying to reduce the lower detection limit. So we have the potential of showing more concentrations. That really the concentration or the detection limit, the method that we're using is really high. So we don't have a lot to show. So those results will be forthcoming as soon as we work that out. This is the seawater data, again, from our deployment sites. So this is all concentrations in the seawater. So here's all of our sites. Here's the month. If it's kind of getting cut off up here, but these are below the detection limit. So here's our 2 that we did detect. And so they're right at that detection limit. Detection limit's 0.1. So we're at 0.1 and 0.11 basically in the Lynn Haven and James sites. So moving on to domoic acid. So this is amnesic shellfish poisoning. Again, you saw maps of Pseudonychia. It's everywhere. We kind of have variable concentrations of Pseudonychia throughout the interior of the bay as well as on the seaside. And this last year, we saw a similar pattern that we've observed the last couple of years where domoic acid is really showing up primarily along the Atlantic. So the white dots, no domoic acid or below detection limit, I guess I should say. And the blue, increasing levels of domoic acid. So if it's blue at all, it's been detected. And then these larger dots, it's over 1 part per billion. The highest was 2.2 part per billion. That was from this data sample here in Rudy's Inlet along the Atlantic Ocean. And then here's a similar situation. So these are cell counts. All of these green means that, here's Pseudonychia cells, but no toxin. The one concentration or the one sample here, this is the Rudy's Inlet, where we have our higher toxin concentration of 2.2. But for the most part, all of the ones that we're seeing are either very, very low or non-detectable pseudonychia, but we're still seeing domoic acid. And so now here's the distribution from our sites here. So here you can see then January through December, and for the most part, again, it's our Eastern Shore sites. So Wise Point through Chincoteague, we're seeing it almost the entire year. So the larger the circle, the higher the concentration. I don't know why March is so good, but you know, no toxin from any of our March samples. But really, toxins, domoic acid at all of these sites throughout the entire year. And then we did see detections in Lynn Haven and James in the water early on in the year, January and February. So now we're switching over to oyster meat, 5 minutes. All right. So lots and lots of what we want to see from public health, empty circles. So these are below the detection limits for domoic acid. And the one site where we did see it in the oysters is the York. And so if you're paying attention, that doesn't seem to make sense because we haven't seen domoic acid here at our site. You know, the same at the Vims Pier. We also weren't seeing it from our other sites really inside the bay as well. So this is January. I went back and looked at the 2022 data, and we had some toxin concentration here as well. And also, or we had samples in the York in 2022. Also, no no pseudonychia and no domoic acid. So I don't know exactly what's going on with that one. Although I am very happy to see this, where we have toxins, they're still not making their way into the oysters on the shore. And we had talked in the past about maybe they were just at different times where the toxin was present part of the time, but the oysters were not pumping. But now we can see like there's toxins throughout the entire year, and it's really not making their way into the oysters, which is good news. But we do have this one. So how do we deal with this sample here? So this, when we put it into the same units, 104 parts per billion, we put our closure criteria in here into the same unit, and you can see, and we're still several orders of magnitude below public health level where we would have a shellfish closure. So I wanted to get that out there. And that's about it. So again, I think we want to continue to focus on this toxin testing and shellfish deployment. There is still a continued need to do phytoplankton analysis. Looking forward to learning more about the planktoscope, seeing how that could maybe incorporated. I'd really like to see more about the SIPs, the new SPAT technology. Maybe that's something we can do as well to try to get some more toxin information. We're looking at this year at putting an additional site on the shore. You can notice that there's nothing on the bay side here, so we want to try to get a site up on the bay side. And then again, doing some additional toxin work with VIMS looking at this OA. Kim's going to be talking about the qPCR data. We still have to figure out kind of some relationship data between the cell counts and the PCR. And then we did get 2 papers out. 2 papers were published this last year. A lot of this toxin data is very similar to what Sarah Peace was able to publish. It came out in Toxicon. And then Margie mentioned too, Jen Wolney has a paper out on Karenia, and there's some of these data in as well. So thank you very much.
Okay, thank you, Todd. Savannah, do we have any online questions?
We do for Margie.
Okay.
Should I ask?
This Margie?
Yeah, that Margie. Margie, Jennifer Salerno wants to know, are these bloom data publicly available? And also, are the water samples preserved or archived?
Oh, the water samples are preserved with Lugol's, and the data, the Bay program data all gets submitted and so is publicly available, and we submit all of our data to VDH as well. The time series site data is not necessarily. Yeah, we keep our mooring data, we upload that into VCUS, and so it's available there, but there's no place to put the cell count data. Yeah, but we're working on getting it published.
Margie. All right, we're gonna go ahead and have our next speaker give her talk. Our hostess with the mostest, Dr. Kim Reese, with the Reese Lab Marine Recap 2023. Kim has been at VIMS for more than 29 years and has served on the HAB task force for more than 21 years. During this time, she and her lab members have been working with the Department of Health to monitor the lower Chesapeake Bay waters for HAB organisms using microscopy and PCR assays. Her lab also conducts molecular genetic studies on aquaculture shellfish species and aquatic pathogens of humans and shellfish. A primary focus of her research is examining the biological impacts of harmful algae on aquatic organisms, a key component in the development and optimization of molecular diagnostic assays for viral, bacterial, and protistan organisms, including the HAB species, in various environmental matrices, including water, sediments, and shellfish tissues. Thank you, Kim.
Thanks, Margaret. Okay, that was whew. All right. Yeah, that I think covered it, most of it. It sounds like Margie and I had the last 24 hours about the same. We had a few illnesses in my lab that kind of disrupted organizing for today, but I think it's all going to come out okay. We're all going to be all right. So yeah, I think Margie gave us a really good background of what I'm going to cover. So some of it I'm going to be able to jump over, and hopefully we'll be able to get to some discussion questions here pretty quickly. This is a picture of Alex Bloom. No, is this Marge or Alex? Do you remember? I think this one's Marge, yeah. Here in the York River that Savannah took from a plane. So this can show you what happens in the late summer here in our York River. So I'm going to focus mainly on the York River because Margie covered kind of what goes on overall in the lower Chesapeake Bay and showed you throughout the year. So she covered this a little bit, but in the typical spring season, this is what we see. We see those heterocapsa blooms, and actually it's already started here for 2024. We're starting to see heterocapsa out there. We often see Procentrum along with that, and indeed we're starting to see a little bit of that as well. And then when we see, as Margie said, when we see Akashiwa, we start to get our radar up and we're looking out for Marge because Marge often follows Akashiwa in our pattern. And Carlodinium, we're always looking out for Carlodinium, partly because my lab has been particularly interested over the years, and we work with the aquaculturists, the shellfish aquaculturists here. And Carlo has often been a problem, especially for the hatcheries. Carlodinium can be a problem. And we've worked with Juliet's lab. Marta has done several studies with Carlodinium. And it's a concern because Carlodinium can be really harmful to shellfish and particularly to the larval stages. So this is an organism that is a concern to the oysters. So 2023, yes, we saw heterocapsa. Savannah Mapes picked it up on the flow cam with about 22,000, almost 23,000 cells per mil of heterocapsa early in 2023. I think that was back in February or very early March. And again, we saw Procentrum also with a qPCR cell count up to about 19,000 cells per mil. And we often see those two together. So we saw large blooms of both of those in the spring of 2023. So we saw these in both the Rappahannock, and this is a satellite image that we got from our colleagues up there at NCOS. And you see here in the James, the upper York, and this is where we were picking up these samples with the really high cell counts of Procentrum and heterocapsa early in 2023. And here up in the Rappahannock, you could see the very dense blooms of heterocapsa and Procentrum. This is March of 20th. So what, and Margie already showed you this, but this is typically what we see, this progression from March to Alex. And we've seen this typical pattern from like 2007 in the York River of Marge to Alex. And as soon as we think we have this figured out, we saw a few years where it wasn't quite like this. But sometimes during these blooms, we see reports of some mortalities. So in 2022, it was a little different. So I decided to go ahead and this year actually graph out some of what happened in 2022. And we had primarily a marge bloom. Most of the river was covered in this very dense marge bloom. But there were these very dense patches of Alex in several places. So I went ahead and graphed the cell counts of some of the Alex. And if you look at it, throughout this marge bloom, we really had dense Alex. The cell counts were really quite high for Alex, even while the marge bloom was going on. But even though you see that the dates were the same, these patches weren't together. Often, sometimes we would get samples where we would have high marge and Alex together, but often you don't see that. You would see dense patches of Alex and dense patches of marge, and you drive the boat and you'd get a sample and you pick it up and it was primarily marge, and then you drive a little further and you pick up a dense patch of one marge, one Alex. And it was very interesting to be out there sampling in 2022. And then 2023 came along and it was almost all marge. We didn't see much of Alex at all. There were just really low cell counts of Alex in 2023. So once you think you have this thing figured out, you don't, or at least it can be frustrating. So these are just some of the aerial pictures. Wolf Vogelbein, our colleague who's now retired, he took some of these aerial photos. This is Alex back in 2016, and this is one of Savannah's pictures of marge in 2022. So this is 2022. This is the York River. This is what marge looked like in 2022 in the York River. You can see it's very dense here, and it was really concentrated here, the qPCR. We really had very dense marge bloom going on. And Alex, again, 2022. And again, these were these patches, but these would be patches that were very dense, but not where marge was located typically. So what happened in 2023? What was the pattern? Well, we first noticed the bloom of marge in the smaller tributaries here, like in Sarah's Creek and in the Perrin River here, off of the main stem of the York. That's where we first got the samples of marge. And that was in late July of 2023. By the first week of August, though, we had samples with greater than 10,000 cells per mil out here in the main stem of the York River. And by the middle of August, we had a sample over on Yorktown side of the York River with over 52,000 cells per mil of marge, and cell counts of over 10,000 cells per mil through the end of August. And the samples with cell counts in the bloom levels, we consider a bloom level of marge over 1,000 cells per mil, held through September 12th, so mid-September. So we had a long marge bloom in 2023 in the York River. And I don't think they saw as much of a bloom, Margie, right, down in the Lafayette James River area. The marge bloom wasn't that sustained in 2023. Is that correct?
Yeah, no, it was.
Yeah. So if we looked over the years, what we've seen is heavy blooms of marge and Alex in 2027. So all of these years, we saw blooms of both of those organisms. No blooms in 14, 18, or 19. Marge only in 10, 11, 21, and 23. Primarily, we saw some Alex around in some of those years, but not. and then a dense bloom with primarily marge, but some dense Alex patches in 2022. So Dante, we need some models to figure all this out, okay? So what we've been doing, and really Gail Scott, sitting in the back hiding there, has been working on this primarily with the Department of Health, trying to develop quantitative PCR assays to, apparently this died. Can you hear? Okay, now it's working. To target these species of human health concern, pseudonitious species, Dinophysis species, and the Amphidomataceae. Did I do that? Amphidomataceae? The Amphidoma species and the Azodinium species, the azosporacid producers that Todd was just talking about. So these are all very important for of human health concern. One of the things that's been interesting is we've been having problems having enough control material for Pseudonychia, but it just so happens that right now, and hopefully Julia, are you going to talk a little bit about the Pseudonychia? You're not going to mention it, okay? Well, we happen to have a source of maybe some control material right now happening because there's a little bit of a Pseudonychia bloom going on, but out there. So maybe we'll have some control material for our Pseudonychia assay. But what we've, for the Pseudonychia assay right now, this is what it looks like for the qPCR results. What you saw earlier from Margie was the visual results. So Todd's working on looking at the data for correlating the visual and the qPCR results here. But these are the results from the qPCR assay targeting the Pseudonychia genus. And what we have here are, you can see that most of the time the cell counts here are, again, not that high based on the qPCR assay. But Gail's been working hard and doing some sequencing to determine which species we have, because it'd be good to maybe the battery's going down on this, I'm not sure. But we're trying to determine what species we have. It'd be nice to be able to narrow it down to have a targeted assay for particular species. But you can see there's quite a collection of species here. Sure what's going on there. So this is the list of species so far that we've been able to identify from the sequencing Gail's done. Here's the Dinophysis again, as Todd pointed out, less than 10 cells per mil. And most of those higher counts are coming again down here in the higher salinity waters on the eastern shore by the qPCR, and out here at the mouth of the Chesapeake Bay. And for the Amphidoma and Azodinium species, we see them all over, as we did with the toxin, as Todd showed with the toxin, you can see some of it all over. The higher levels by the qPCR counts are here, Mobjack Bay, and maybe over here on the eastern shore and up here in the northern neck area on the Potomac. But again, these levels are very low. We're just looking at much less than 100 cells per mil. So very low levels of these species as well. But again, we're interested in identifying these species and working with a colleague in Europe also to possibly hopefully get a culture of these organisms established and do some species identification. One of the things that came out of listening to what our colleagues at the Maryland HAB task force meeting was whether we should add some of the Karenia qPCR assays, and maybe that's something we want to discuss because it seems like maybe that's something that would be important given what's being observed. So the spring blooms were dominated by, and still are dominated by, Heterocapsa and Prorocentrum, as we would expect. March summer bloom in 2023 persisted for several weeks in New York and in 2022. And assays for the human toxic species are still being optimized for Chesapeake Bay species. And we need to do that study looking at the correlations between the qPCR assays, the visual counts, and the toxin levels. And there are several different pseudonischous species apparently in the region, and we need to look at which ones are the important ones to target, which ones are the human health concerns, which ones are the toxin producers. Do we need to add the Karenia species? I think those are the important questions we need to address right now.
Thank you, Kim. All right, our next presenter is Lindia Branch, Statistical Analyst, Virginia Department of Health, Office of Environmental Health Services. She's going to be presenting the Cyanobacteria Advisory Recap for 2023. Lindia has worked for VDH for 2 years and migrated from local health to Division of Shellfish Safety and Waterborne Hazards in March of 2023. Lindia has a diverse background spanning public and environmental health with experience in data management, and she is a joy to work with. Thank you, Lindia.
Good morning. So today we're going to cover the Cyanobacteria Advisory Summary for the freshwater season for 2023, as well as HAB meetings and other efforts, and then the upcoming 2024 HAB program priority projects. So for the 2023 season, we received a total of 137 HAB reports online. We had 2 major water body advisory events in 2023 at Lake Anna and Smith Mountain Lake. Lake Anna was under advisory for all reaches for a total of 122 days. And then Smith Mountain Lake had an advisory in the Blackwater River arm that lasted a total of 78 days. I also want to point out that the advisories issued for both water bodies, they were due to cyanobacteria cell density counts rather than high concentrations of cyanotoxins. So we only observed one site at Smith Mountain Lake where there were toxins above 1 parts per billion, but they were well below the advisory thresholds. And then over the season, there were also some, a few investigations that occurred in the Valley area at the North Fork and South Fork Shenandoah River, but there were no significant blooms that were observed at that time. So no MAT alerts were issued for 2023. So for Lake Anna, compared to 2018, 2020, or backdated to 2018, 2023 was the second worst season on record. 2021 had a total of 123 days, whereas 2023 had a total of 122 days. And then what's listed in the map is the issued advisory that took place. The issued advisory, the initial advisory was issued on June 29th, lifted on August 7th. Try this one. It was lifted on August 7th. Lake Anna is a recreational lake that's about 13,000 acres in size. And the blooms predominantly occur above the Route 208 on the lake. We received a total of 25 online HAB reports for the 2023 season for Lake Anna. A total of 49 samples were collected and analyzed. And then also, the advisory that took place from June 29th to August 7th occurred at the Upper Pamunkey. There were several reports for the lower lake in the lower part of the lake in August. We were unable to investigate that, and that is Old Burris Mill, pictured on August 30th, 2023. And we also received a few reports in the tributaries from the Lake Anna Civic Association as well as citizens. So there were a few reports in the waste heat treatment facility lagoons. These incidents were reported to Dominion Energy and they were investigated by Dominion, and there was no observance of a bloom that occurred. So no advisory was issued for those parts of the lakes as well. So for the initial advisory issued at the Upper Pamunkey that was lifted on August 7th, There was another advisory that was issued that same day for the Upper North Anna branch, and that one was not lifted until November 1st. The Middle North Anna branch also was under advisory from August 18th to October 16th. And there was a second advisory issued for the Upper Pamunkey that took place from September 22nd through November 1st. So all of the advisories that were issued on November 1st, they were due to the conclusion of the 2023 HAB response season, even though blooms could be ongoing through the winter. I want to point your attention to this blue box here. This gives you a total or the highest cell counts that were seen during the sample collections for the Upper North Anna, the Middle North Anna, as well as the Upper Pamunkey. And there was one site that, well, all three segments, microcystin toxin was detected on October 10th at all three sites, but they were well below the advisory threshold limits. And then the map shows the worst extent of the advisory that took place in September. So moving on to Smith Mountain Lake, I want to give a little background. Smith Mountain Lake is a 35,000 acre in size. And then it also has some drinking water intakes on the Blackwater River arm. It is formed from the confluence of the Roanoke River and the Blackwater River. And then also for 2020 HAB or coordination efforts took place with the Smith Mountain Lake Civic Association as well as Ferrum. And this was coordination for response activities. So from 2020 to 2021, both years there were short bloom events, but they dispersed within a few days. So no advisories were issued, and this took place in late May and early June. So our Ferrum partners, they confirmed the presence of cyanobacteria species that's capable of producing toxins during these short events. However, we were unable to collect samples and get them ID'd and get them to our labs for ID enumeration and toxin analysis. Between 2020 and 2021, online HAB reports have been received annually, and they were in the Blackwater River arm. However, there were no reports in the vicinity of the state park or the Roanoke River arm. So 2023 is the first year that an advisory was issued on the lake. I want to point out that there were a total of 57 HAB reports received for Smith Mountain Lake. There were 29 samples that were collected and analyzed from the water body. There was an initial bloom reports that occurred on between May 21st to the 22nd. We were able to get out, or DEQ was able to get out and collect samples on the 23rd. And the sample results determined that no advisory was needed for that time period. The following week, we received several additional reports for the Blackwater River arm. Sample collection was conducted and an advisory was issued on June 6th, and it was for the entire Blackwater River arm portion of the lake. So sample collections occurred at Anthony Ford and Kemp Ford, as well as Virginia Key. Two of those locations, which was Kemp Ford and Virginia Key, there were sample results that were between 100 to 200,000 cells per milliliters. And then at the third location, which was Anthony Ford, there was over 1 million cells per mil in that area, as well as antitoxin A and microcystin toxins were detected, but they were below 1 parts per billion. All sites were collected along the shoreline versus in the mid-lake channel. And that's significant because it takes a greater effort to collect along the shoreline. And that was something that was done differently than in the past because most samples are taken by boat. So scums and visual blooms, they can accumulate along the shoreline versus in the mid-lake channels. For Smith Mountain Lake, we had a total of 4 health complaints. We were able to investigate 2 of those. And these all occurred, or exposure occurred during the month of June. And exposures was said to have occurred the week prior. So of the 2 that we were able to investigate, the symptoms included vomiting, diarrhea, nausea, sensation of sunburn in the absence of an actual sunburn, as well as skin rash and swelling and blisters. In mid-June, so about June 11th through the 19th, we received additional complaints in the upper and mid Roanoke River arm tributaries of the lake. They were predominantly near Betty and Becky's Creek, just north of the state park location. On June 15th, about 80% of those complaints that were received, we were unable to investigate. So we asked our partners to voluntarily place signage to beware of algae blooms. And this was due to weather. So we weren't able to get out until late June to collect samples. And no samples were able to be collected in Roanoke River. So additional sampling in late June of the Blackwater River arm allowed us to lift the advisory from, instead of from here, we were able to reduce it back to this area of Gills Creek. And this occurred on July 7th. And then the remaining sites, they weren't able to be lifted until August 24th. So for other HAB projects, in order to streamline efforts and establish greater consistency, VDH and DEQ did execute a collaborative MOU that outlines the agency's respective roles and responsibilities as HAB task force co-leads. And this requires routine, high-level meetings to ensure agency goals as well as priorities are being maintained. And then this will be discussed a little later. There's a SPAT project on the Shenandoah River for waterworks intakes by Samantha Mooney, who was a PhD student under Scott Glauberman. And this wrapped its inaugural run with the Berryville SPAT preliminary results. And the results indicated that a very low but detectable levels of neurotoxins from deployments which ran from August to October 2023. And Samantha Mooney will go over that. And then there was also a project with Gold Standard Diagnostics and ICPRB that wrapped its second year in 2023 with mat survey collection analysis, with trial runs and feedback to test strips for novel macroation device to aid in the extraction process of cyanobacteria mats. And this will be discussed later as well. We also completed and posted the private water body guidance, and that's located on our Swim Healthy VA site under HABs. And then we also launched a new tracking tool to aid us in the review and management of online HAB reports that were received throughout the year. So for upcoming projects, we want to improve the flow and collaboration of sample and shipment. So we're currently evaluating ways that we can make the process better and things that we can do to aid our labs. We're also consulting with GMU academic partners to update our PTOX taxa list for possible additions to cyanobacteria mat groups. And then we also intend to create more transparency with the public. So we will be adding all HAB reports received to our HAB map. So look for a change in our key for that coming soon. And then we also intend to continually support ongoing projects, which include our GMU partnerships and ongoing studies of Lake Anna and the Shenandoah River, as set to complete between 2025 and 2026. So just want to say.
Great. Thank you, Lydia. All right. We do have time for questions. Do we have any questions in the room?
Question for Kim, actually. Mentioned the student bloom going on. I don't know if that's the one that we were talking about before. We did get samples. We had reports of a pseudonytzia blooming near Gwynn's Island. And I was able to get some samples from that. Jackie got them right near the end of January, and I guess we finished them on Thursday. So they all came back below detection limit for domoic acid as well. So again, that's similar to what we've seen before. And those were really high. I don't know if you have counts, but we had, in the high hundreds of cells per mil, and had heard reports maybe of even higher concentrations.
Well, I don't know if Savannah's counted it.
Yeah.
Have you counted the cells
On the bloom, did you count that yet?
No, just that there's a lot of cells out there right now.
Yeah.
We had seen, that was the highest concentration that we had seen, which is why we wanted to get some domoic acid results, and
Yeah.
those were all again negative. So
That's good that's great for the domoic acid. I'm glad.
Very hard to count.
Oh, absolutely. Oh, I agree. Yes.
maybe even impossibly, but
Trying to get a standard for the cupid stuff.
Yeah.
working on it.
Okay. Cool. Thanks.
Thank you. That's good.
Any other questions in the room?
Question up there.
Yeah.
While Roger, there's also a header capsule that's been going on in the Elizabeth River.
I was wondering if there is an interest or plan to start sampling with higher frequency during the November to May period. Things like anatoxin occur oftentimes in cooler weather and can be missed, I would think, in many systems. Thank you. The question.
We do have a question from the Zoom folks. What were the dominant species in Lake Anna and Smith Mountain Lake when advisories were issued? Do you remember, Todd? I don't remember. I can send it to you, Kathy.
Yeah, in Smith Mountain, aphanizomenon and Delicospermum were the two most common when we were picking them up in those bright blue colors you saw. So those were the two dominant species. And those aphanizomenon hadn't been found in any numbers for years there. Bedford had a problem near their water plant with some runoff from hydroseeding, but years, that was early. So that was new. And that's why Riley was talking about, we added those species because they were new this year. Of course, Delicospermum was Anabina originally, Circinalis.
I don't know, Paul. It's not the answer, but the main reason we're working in the warmer months is that it's largely recreational. So that the warm weather sampling is largely, when the recreation is going on for swimming. The fact that there's drinking water implications would change that. So that I know there is, I don't know if Bob, you want to say anything about drinking water, or that there is some level of surveillance that goes on throughout the year, by the drinking water plants as well.
Hold on, Adam. Hold on. Resist the urge. Wait.
Question. This is going back to Acidinium. What's the seasonality with that?
I don't think we know very much on that yet. That's another one where there's not a commercial test for the azosporacid, so we don't have routine azosporacid testing. The only azosporacid testing was on the CB Tox project. I know, so we had really low concentrations there. We could look at some of those figures that Michelle had. I can't remember azosporacid seasonality.
The toxins were persistent all across the year.
All right. And the second part to that, and this is kind of where I'm going with it with the health implications, is we haven't really seen any illnesses, I think, in the US because of it, just over in Scotland, right? But what's interesting to me is just thinking about why we might not see it, because I think the symptoms are very consistent with Vibrio parahemolyticus. So it's kind of in that same time frame. Like that might be one of the things that we see where we have very similar illnesses for Vibrio, but they don't necessarily test positive when we get a COVID report or look at anything from hospitals with that. So just food for thought.
Great point. And I just gave my lecture on azosporacid, so this is fresh in my head. But the way that azospораcids were even found was because there was a bunch of poisonings that look similar to what we're describing, but not in the US. But it turned out that it was actually, they had first started looking for dinophysis toxins. And then when they didn't find oxalic acid and dinophysis toxins or any of their derivatives, they started looking for something else. And that is when they found azospораcids. But yes, as I know of, there is no reported illnesses specifically due to Aspergillus yet in the US. So it's an upcoming emerging species of concern. Does that answer some of? Yeah.
Yeah, absolutely. We can talk more about it, but it's gonna be interesting because it's just likely to be covered up with things that we do see already, as opposed to other countries where they aren't really seeing those VP cases.
Kathy Wozniak, I think I just remembered Lake Anna was Raphidiopsis. That's kind of been the dominant in Lake Anna. We got some more online questions. Go ahead.
You get to read them. Okay. Can you hear me on this? No.
So Kathy said, thank you. We have had high microcystin with aphanizomenon in some of the lakes in Maryland. And then Jeff said that DEQ is sampling during the colder months for the Lake Anna study, and ODU is also looking at them. Kathy said, thanks.
All right. Thanks everyone for the great commentary and questions. We're going to go ahead and move on to our next speaker who is speaking remotely. Boris Polyak. He is going to provide a presentation on the field strip test for benthic mats. Boris, are you able to bring up your presentation?
Is it not up?
We're not seeing it yet, but we can't hear you.
I shared it and it appears on my screen as shared. I'm not able to turn on my camera.
All right, there we go. All right, let me just get through your introduction real quick, Boris.
Okay.
A biotechnology engineer with over 20 years of experience in biological sensing, immunoassays, and lateral flow based rapid detection systems. Boris leads gold standard diagnostics consortium R&D on strategic and operational activities focusing on product development and commercialization, product support, and contract R&D. He is responsible for building an ecosystem of cross-industry, academic, and government alliances and partnerships while engaging in business transformation to improve the spectrum and quality of environmental, food, and life sciences test systems. Thank you, Boris.
Thank you, Margaret, for the introduction, and also thank you for the opportunity to talk to all of you today about this collaborative project. Lisa Camp is our senior scientist who was the major driver behind the planning, execution of the experimental work and the data analysis. Other individuals also contributed to the development and commercialization of this product. Fernando Rubio is the founder of Abraxis, which is known today Gold Standard Diagnostics. Lori Fields is our director of water quality portfolio and marketing, and Stacy Hirgzel is our product manager. I would like to also thank the team of Virginia Department of Health, Todd, Olivia, Jim, Margaret, and Daniel for supporting this project and great coordination. Also, I would like to thank the teams on the Interstate Commission on the Potomac River Basin, Charlie O'Brien and Gordon Mike Seltman, for collecting samples and running the beta test on site. And also I would like to thank Andy Chapman and Amanda Foss from the Green Water Laboratories for doing taxonomic analysis. So speaking of how would you approach, Abraxis and today Gold Standard Diagnostics is known for development and manufacturing of a variety of cyanobacterial assays, including all these toxins on the left side in the laboratory ELISA format, as well as we have several rapid test strip kits for microcystin, cyanotoxin A, cylindrous primopsin, saxitoxin, and currently we're working on development of the domoic acid and beta toxin assays. And those assays were mainly designed for planktonic forms of cyanobacteria or extracted toxins from those planktonic forms. And they are not really suited for dealing with thick benthic mat samples that is currently big interest in the field. And we know that some cyanobacterial species can release toxins into the water without cell rupture or death. However, most of the bioavailable toxins are intercellular. They accumulate within the cyanobacterial mats. And in many cases, the release of those toxins in an algal bloom into the surrounding water occurs during the cell death. So when we speak about testing of benthic mat samples in the field, you obviously need a rapid strip test, which we and others offer today. But another component of this testing is the efficient field-based extraction method. So the traditional freeze-thaw methodologies used in the lab are not useful for benthic mats, are more suited for planktonic forms. And usually people perform mechanical homogenization using commercial bead rupture methodologies. So we asked the question, what can be done in the field? How the sample can be processed in the field on-site and tested immediately using the rapid strip test. And we wanted to investigate and corroborate the possibility of using the mechanical homogenization using just a very simple device, which is shown here. It's a tissue grinder tube with a pestle that can be used to mechanically process and homogenize your sample on site. Here are some samples of the benthic mats that were collected by the ICPRB team. And when we blot those samples, we observed colors that varied from bright green to blue-green, dark green, brown, and even black. So these samples consisted of multiple cell types of algae in each sample, and they were feathery, well-defined strands and gelatinous to touch morphologically. So before approaching the field to test alpha and beta testing, we wanted to validate the mechanical homogenization method. And in this study, we worked with benthic mats that were collected in August and September 2022. We used ELISA to compare between the two methods. And we selected a negative sample mat that was spiked with anatoxin A, microcystin, saxitoxin, and cylindrospermopsin. And we determined the recoveries of these toxins. And this graph shows excellent correlation between the two methods with a coefficient of correlation above 90%. So this is a typical flow chart for this product. Similarities and requirements for toxin stability and assay tolerances enabled us processing samples in grouped extractions. So you can process microcystins and cylindrospermopsin, in anatoxin A and saxitoxin in the group extractions. And let me just zoom in here. I will walk you through the extraction procedure. So the first step, you tear your tissue grinder tube, and you remove the sample from the sample collection vial. You measure 1/4 gram of your sample, and then you add 0.5 milliliter of the benthic mat extraction solution. And the next step, you perform this manual mechanical homogenization for 4 minutes. And then you transfer sample extract to a dilution vial A, which contains 2 milliliter of the same extract solution that was used during the tissue grinding. You shake, you let it settle for 10 minutes, and then you prepare another dilution vial B with 9 milliliter of distilled water for the further dilution. You transfer from the upper sample part of the vial into this vial, and then you mix it again, shake, let it settle for additional 10 minutes. And again, you sample from the upper solution of the soil and conduct your test. So before diving into the results, I would like to shortly talk about the principle of the lateral flow test. This is a competitive assay. So the central component of this assay is the antibody against your toxin, and this antibody is conjugated to a gold nanoparticle, which is your detecting element. So depending on the concentration of the toxin, you may obtain a range of either fully blocked antibodies, partially blocked, or not blocked at all. And when you apply the sample to the dipsticks, the samples start flowing along the strip. And if we zoom in into the nitrocellulose component of the test, we have another element that is immobilized on the test line. And this element is a toxin conjugated to a protein carrier. And practically your antibody here is competing for the toxin in the solution with the toxin immobilized on the test line. And when this interaction is complete, antibodies continue flowing along this strip. And those antibodies linked to gold nanoparticles are captured by the reagent deposited on the control line. And this reagent is an antibody that can recognize the antitoxin antibody to demonstrate that your test was conducted properly. Interpretation of the results is shown here. You can observe negative results by two solid lines of similar intensity. And when the toxin present a certain concentration, the test line starts to fade out and can completely get eliminated in the case of very high concentrations of the toxin. And you can do the interpretation either by visual method or using a reader, which can give you more quantitative representation. So this is the panel of results for anatoxin A. You can see here the line intensities for the control line, test line, the ratio, the final result after factoring the dilution, and we also compared this with ELISA. And we tested several samples that were collected on August 24th and August 30th. And we were struck to see relatively high levels of anatoxin A, which correlated well between the strip and the ELISA. The sample collected later on August 30th also showed some presence of anatoxin A, but at much lower levels. And qualitatively, you can see here stronger detection of anatoxin A, with test line completely disappeared, with some test line at much lower intensities for these two samples. And on August 30th, you can see one sample that was non-detect, two lines of a similar intensity, and the test line started to fade out for these low anatoxin A containing samples. To rule out potential matrix interferences, we conducted a dilution study, and we compared the commercial method with a tissue grinder for the field extraction. And you can see that the recoveries relatively to the undiluted sample are independent of dilution, which indicates no matrix interference in this testing. So this panel summarizes saxitoxin results. We only tested one sample collected on August 24th. We detected relatively low levels of saxitoxin, approximately 10 to 20 parts per billion. These results correlated with ELISA, and these are the qualitative images showing the non-detect sample, and these are low positive samples. Again, dilution studies indicated no matrix interference as recoveries were independent of dilution. For microsystems, samples on August 24th were all negative. We only detected a low positive sample in the later collection date. And again, the dilution studies didn't indicate any matrix interference. And finally, cylinder spermopsin analysis revealed no toxin presence at the detection levels of both lateral flow system and the ELISA. And we also conducted dilution studies to show no matrix interference. We have some data on the taxonomic identification here that was provided by Green Water Laboratory. And the most abundant species, and I'm going to focus on potentially toxigenic species first, was the Microcera or formerly Lingbia volii. These species are known to produce cylinder spermopsins and the forms of cylinder spermopsins, saxitoxins, and the Lingbia volii toxins. Other species were oscillatory and formicium. They are known to produce microsystems, anatoxin A, homonatoxin A, and cylinder spermopsins. We also detected Lingbia species in a sample that was collected on August 24th. And last toxigenic species were Anagnostinema or formerly Geitlerimema and Jaginema species, which also were reported to produce the saxitoxins, goniotoxins, microsystems, and anatoxin A. Other non-toxigenic algal groups included filamentous green algae and diatoms. Here are additional pictures from other dates and the samples that contained very same species. You can see the same faces here, and the non-toxigenic were Cladophora glamerata, Spiragyra, and the conjugated form of Spiragyra species. And diatoms were again the non-toxigenic part. Here is another example of benthic mats from the later collection date, pretty much same organisms, potentially toxigenic, and the non-toxigenic are same groups of filamentous green algae and diatoms. And this is example of the third sample. The Microcera volii was the most abundant as the toxigenic species, and the Cladophora glamerata was also most abundant among the non-toxigenic species. So this slide summarizes all non-toxigenic species. For those that are interested to review, this presentation will be posted on the website. And finally, to conclude, when you're dealing with benthic mats, it's important to keep in mind that these are very frequent non-homogeneous samples. They require extra care when you work with them and collect them at the site and then prepare for the testing. So you have to be really careful, and they use several spots for sampling and at the site, and also when you use your sample for sample preparation. We demonstrated that manual tissue grinders can be used in the field testing, and the results of extractions using those devices were comparable with the commercial bead-based methods. Our studies indicated lack of matrix interference, so we believe that the toxins that we detected by both TRIPS and ELISA were true representations of these toxins. We also observed very strong correlation between the methods for all toxins and all samples, despite some sensitivity differences between the strip test and ELISA. And finally, the potentially toxigenic species correlated with the detected anatoxin A and saxitoxin in the collected samples. Here I provide our contact information and also a reference for the Green Water Laboratory's potentially toxigenic cyanobacterial list that we can find on the research gate. And thank you for your attention.
Thank you, Boris. Do we have any questions from the meeting? Do we have any questions in the room?
Hi, Boris. This is Chris Jones from George Mason. So your tissue grinders looked a little different than what I normally think of as tissue grinders. The ones that I'm familiar with. It's not working. Maybe I'm not supposed to turn it on. All right. The ones that your tissue grinders look a little different than the ones I'm used to. They looked like they were conical on the bottom and they had a cap. I mean, they almost look plastic to me. Can you elaborate on those? Because I'm familiar with the glass tissue grinders with the glass pestle.
Yeah, those are tissue grinders that they made of plastic, and the tissue grinder tube is a standard 1.5 milliliter Eppendorf tube. I think those, maybe Lisa can help me here, those made of a plastic type that absorb the toxins the least, similarly to glass, because this is a crucial property of the material that you use in those systems. I think these are just very small and disposable tissue grinders that we found commercially available, and we conducted a study where we demonstrated that there is no absorption of toxins on the plastic walls of this device, and this device can be used comparable to glass material, and because this is plastic, it's cheaper and disposable, so this was the main rationale behind the use of those devices. If Lisa has anything to add, please feel free.
Thank you, Boris.
We do have a couple online questions.
Okay, I think we have time for one.
Okay.
Wait, where are they? Right here?
Mhm.
All right. Do you know the station locations?
I
And
don't,
go ahead.
yeah, I don't know the station location. The samples were collected by the ICPRB team, so they have this information.
Yeah, so Margaret, they were near Strasburg Park.
They were near Strasburg Park
On the North Fork.
on the North Fork. We can share that with you later, Kathy. We're gonna go ahead and move on to our next presenter. And we'll try to come back to these questions at the end, Kathy. Okay, so our next presenter is also going to be presenting remotely. Samantha Moni, PhD student at George Mason University, GMU SPAC collab with Shenandoah River Water Utilities in 2023. She's an environmental science and public policy PhD student at George Mason University, studying benthic cyanobacteria and toxin production within the Potomac River Basin and its tributary, the Shenandoah River. Her work involves investigating regional benthic have a current seasonality and toxin production in order to build an initial framework for predicting how shifts in regional water quality climate policy will shape the future impact of benthic HABs on environmental and public health. Sam, are you with us?
Yes, hello, good morning.
Hear you loud and clear. Go right ahead.
Okay, all right, perfect. Well, thank you so much, Margaret. Hi, my name is Samantha. As Margaret said, I'm a PhD student at George Mason University, where I am studying benthic cyanobacteria and toxin production within the Potomac River Basin and the Shenandoah River. And I'm here to present to you all about a wonderful collaboration that I was able to do in 2023 with some Shenandoah water utilities and deploying some spats throughout the Shenandoah River. So to begin with, this is just going to be a little bit of review, I'm sure, for a lot of folks, but just to touch over, spats just stand for solid phase adsorption toxin testing. And what they are is they're a wonderful passive sampling device that is essentially provides an in situ assessment, and it's a monitoring tool as well for cyanotoxin presence in water bodies. It's really great because it can be complementary to a lot of time series projects that are ongoing. For example, so like some of my other projects, it's very complementary to the work that I'm doing. And most importantly, it can be used jointly with traditional sampling methods like grab samples, so discrete water samples, or in my case, benthic mat samples. And it is a great indicator of cyanotoxin prevalence and persistence as well. SPATs essentially are a time integrated measurement of dissolved toxin presence because, again, they are passively sampling. And I think one of the biggest positives of SPATs is that they provide continuous toxin detection. So instead of having to rely on just discrete water samples, which just gives us a snapshot of toxin presence at any given time, this gives us a better idea of toxin presence throughout a series of deployment times. And this can be really helpful in capturing toxin pulses, especially with discrete water samples. That is kind of one of the biggest downsides, is it's just providing a snapshot. And so let's say a toxin pulse had happened a day or two prior, and we had a major precipitation event that washed it out. We wouldn't really know if we were to just take a discrete sample following that event. So it's very helpful with that. And it can also be great in illustrating certain environmental drivers that might be causing toxin presence within a certain water body. And it's very highly sensitive, even in low ambient toxin concentrations. And so this is a great thing where SPATs are able to show toxin presence where other analyses might not show them present at any given time. And then over here on the lower right-hand corner, you will see an example of a SPAT device. I call it a tea bag because that's essentially what it looks like, where it is just a specialized type of mesh with an absorbent resin, these microbead resins within it, and those absorb the toxins, and then I extract them from the resin and do my toxin analyses. So that's just a brief overview of SPATs. Sorry, and it's great because it's used for a wide range of cyanotoxins, which is fabulous. So the advantages of SPATs, especially when it comes to their use with public water utilities, is their general ease of use. So one, they can be used in all water body types from marine, brackish, and freshwater. And it is material-wise very low cost of materials, which I'm sure most of us that never happens when it comes to a lot of our lab work and a lot of the materials we have to work with. So low cost is definitely an added bonus. And I found that it can be made in batches, which is really helpful when working with public utilities, because I can make essentially a whole bunch of SPATs at a time, and then I can drop them off at these different public utilities. So they don't need to make them. I don't need to, drive over there every week to keep on dropping off new SPAT samplers. And so it's just really nice that you can kind of make them in bulk. They are also very easy for deployment and retrieval. So you'll see over here on the right this series of photos. This is the general setup of my SPAT devices that I used for this project, where the SPAT device, because it's enclosed in mesh, it can be quite fragile. And so I created these protective wire holders over here to insert the SPAT device in there. You close it up, and then you attach it I use zip ties to a structure or a weighted line. And then here at the bottom photo, you can see I have one attached to a PVC pipe that I had kind of pounded into the sediment there. And you have very flexible deployment periods, so it can be anywhere from 1 week to a month. And another bonus that I found with working with the public utilities is that there are very simple storage requirements. So it doesn't require anything fancy when it comes to storage. So pre-deployment, I would drop off SPATs and they can be refrigerated. And these SPATs can be stable for up to 3 months, or Josh previously presented showed that it can be up to 4 months where they can be stable. And so they can just be stored in any old fridge, doesn't require anything fancy. And then post-retrieval, they just need to be frozen at -4 degrees Celsius. And again, nothing special with that. Any old freezer will do, which is, I think, very, very helpful. Unfortunately, the one major disadvantage of working with spats is they only measure dissolved toxins. They don't measure total toxins because that would require cell lysis. So that is one caveat, but overall, I think there's a lot of advantages. So a brief project overview of my spat work with the public water utilities. The problem is we've been seeing toxic benthic HAB proliferations within the Shenandoah River, specifically the North Fork, where we had that really big bloom event in 2021. And so we are looking to kind of gather more information, and spats just provide another piece of the puzzle in trying to figure out what's going on over there. And the general purpose of this project was to provide toxin data from my passive samplers to complement previous and ongoing discrete sampling events. And my partners with this, so my partners were Margaret and Todd. So overall the VDH, and we had the Office of Environmental Health Services and their Division of Shellfish Safety and Waterborne Hazards. We worked with some Office of Drinking Water field offices, and of course, some public water utilities that were located along the North Fork Shenandoah River. The reasoning for partnering with the public water utilities along the North Fork is that it allows me to expand my field design, and in doing so, I can have deployment locations at ODW drinking water intake points and areas where there are benthic mats or there are known toxin hotspots. So again, just allows us to expand our field design and get a much better picture of what we're looking at. Some of the deliverables that we're hoping to get from this project is the overall dissemination of any data, results, and technical or non-technical interpretations from me to provide for the VDH, local water body managers, these public utilities, and other stakeholders within the overall Potomac River Basin and along the Shenandoah River. And then outcomes, we're hoping to use the deliverables that I've been able to provide to help revise public health messaging for benthic algal mats, and also provide enhanced toxin information for drinking water treatment options as well. So that's just a brief overview of the kind of overall reasoning of why we decided to go ahead with this project. And so now on to the actual collaboration itself. So this collaboration occurred in 2023, and I like to call this a pilot study of sorts, where this was 2023 was just us kind of, we've never done this before, and just trying to see what works, what didn't work, you know, ease of use, how, you know, water utilities, the ease of like them deploying, or if it would be too difficult. So overall, I would just like to really point out that 2023 was kind of the pilot study. So I was able to work with the town of Berryville Waterworks with their at their Roy E. Potts Waterworks treatment or water treatment facility, and we were able to go to the treatment facility and go to the drinking water intake site, which was very close to the facility. And what we would do is, working with one of the waterworks operators, once a week, we would have him go down and deploy 2 spats, so just replicates, deploy them at this drinking water intake site once per week. And we deployed them from August to October 2023. The reason why we did August to October is just kind of getting all of the logistics of the project figured out, getting supplies, getting everything ready. We were not able to get all of that done until August. So I would like to say I do know that this is only kind of, you know, half or a third of normal bloom season and sampling season, but that was just primarily due to logistics. And at this intake site, it was very easy. We just identified, it was essentially a concrete block that we were able to attach a line to, a small rope, you might say, and just hang those spat devices from that rope and keep them continuously submerged in the water for a week. Once a week was up, one of the operators would go down to the intake site, take those spats, store them in a freezer, and then deploy 2 new spats. And again, this was ongoing until the end of October 2023. So some of my spat results, first and foremost, for my toxin extractions, the toxins that we primarily looked at were saxitoxin, anatoxin A, cylindrospermopsin, and then microcystin and nodularins. For my, I didn't want to go in depth into my toxin extraction methods. If anybody is curious or would like more information on that, please do not hesitate to contact me and reach out, but I won't really be going into the specifics of that today. My analysis methods, I used an ELISA, enzyme-linked immunosorbent assay from Gold Standard Diagnostics. I used their kits for my work. Typically, LCMS is kind of preferred to have alongside the ELISA. So I would ideally have liked to have both ELISA and LCMS results as well. But just time permitting, we weren't able to do that. But moving forward, that is going to be the plan, to have both of those analysis methods. So looking at detection thresholds, again, there's detection thresholds according to each of the toxin kits. So saxitoxin, we'd be looking at about 0.02 parts per billion is the detection threshold. Cylindrospermopsin was about 0.05, and then anatoxin, microcystin, and nodularins was about 0.15 parts per billion. So looking at those detection thresholds, my actual results for my spat toxin extractions, we didn't really get a lot during this kind of pilot season. But again, with this, I am not fully surprised, just because by this point, again, talking with the ICPRB and a couple other folks, we weren't really seeing a lot of benthic mat growth. And so I would be very surprised if my results had shown otherwise, because that would have kind of gone against everything else that we've been seeing along the North Fork. So with saxitoxin, my results, I found trace amounts in October in some of my samplers. And so when I say trace amounts, I just mean like just above the detection threshold. So for example, with my saxitoxins, I had a couple of my replicates give me about 0.021 and about 0.023 parts per billion. So again, just barely above that 0.02 threshold. Anatoxin A, there were trace amounts in September, but again, amounts were just barely over the detection threshold. So nothing too crazy. And then cylindrospermopsin and then the microcystin and nodularin test, I did not get any toxin detection for any of those. But again, I would like to remind that I was not able to deploy these until August into October. So typically not during the peak bloom period or during the peak times when we would typically see major toxin pulses. So this kind of keeps in track with what I was expecting anyways. So with these results from my 2023 pilot study, what I am hoping to do is I have been working with Margaret and all of these different drinking water utilities to create a much larger 2024 SPAT collaboration project with these water utilities. So we are hoping that everything's keeping in line. Fingers crossed, knock on wood, don't want to curse anything, but everything is, full steam ahead so far. And we are looking to begin deploying starting this April of 2024 and looking to deploy into October or November of 2024. Again, we have been seeing some benthic growth that has been continuing into the later seasons. So, for example, as was previously mentioned, anatoxin can oftentimes be detected later on in the season. So that's why I'm hoping for some later deployment periods. But that's all going to be kind of based on what we're observing at the time. It's fantastic. After my 2023 pilot project, my participating localities and their public water utilities that will be participating in the 2024 season, I am thrilled to say that Berryville will be coming back and participating, but we will also be doing work with Woodstock, Strasburg, Front Royal, and Winchester. And a massive round of applause to Margaret for being incredible and helping kind of bring all of these different localities and all of these pieces together. So this 2024 project, hoping to get a lot more data at a lot more sites to hopefully maybe next year come back and present and give you all a much larger picture of the results from the SPAT project. I would also like to point out that this SPAT project is a part of my larger dissertation work, and it is going to be complementary to work that I am going to be doing at other sites along the Shenandoah, and that is going to be done. I'll be partnering with the ICPRB, where they'll be deploying some of my SPATs at all of the different sample sites that they are going to be looking at, with that USGS, ICPRB, and GMU Shenandoah project that Lex Berger previously mentioned that George Mason and the Potomac Environmental Research and Education Center is a part of. My anticipated work moving forward, I plan to do continuous toxin extraction and analysis throughout 2024 to 2025. Generally extracting toxins from the resin, it's pretty simple. It can just be time consuming. And so that's why I say into 2025 is, you never know what's going to happen. And so I might still have some ongoing analysis through 2025 from the 2024 field season. I also plan on investigating potential environmental trends related to the toxins that I'm seeing and the concentrations that I'm seeing in my SPATs by using temperature and water flow data sets and comparing them to see if there are any correlations between those two variables and the toxins in ambient water. And my findings are going to contribute to my overall larger dissertation project, investigating benthic C-HAB occurrence and toxin production dynamics within the Potomac River Basin. So I'm hoping that all of this work I'll be able to give you all a much broader picture of benthic cyanobacteria and their toxin production capabilities. I think that SPATs are just a wonderful kind of missing piece of the puzzle that will give us a much better idea of what we're dealing with in the Shenandoah River and why we've seen, kind of this very recent kind of, explosion of benthic C-HAB occurrence within there.
Thank you, Sam. Could you please wrap your talk?
Yep. So I just wanted to say special thanks to Margaret and Todd at the VDH. Without Margaret, none of this would have come together. She has been my saving grace with this. Jean Petty was the deputy town manager at Berryville who helped organize some of this. Warren McCormick from the Berryville Water Treatment Plant, he was the operator that was my partner in crime in all of this. Dr. Scott Glaberman from my lab, and also Leah Gibla-Smith, who is in Margie's ODU phytoplankton lab, for answering all of my numerous questions, and everybody else involved. So thank you very much.
Thank you, Sam. Great. Our next speaker is the first of our second student lightning talk sessions. Lily Bloom is here. She's going to be presenting metagenomic insights into microbial stratification during a harmful algal bloom event in the York River estuary. She got her undergraduate degree from Cardiff University in the UK in marine geography and joined VIMS in the summer of 2021 and now a PhD candidate in her third year. She is co-advised by Kim and Dr. BK Song. She studies HAB dynamics and their associations with aquatic microbial communities.
Sorry, this is taking me a while. Can everyone hear me? It works, right? Perfect. All right. Thank you so much, Margaret, for this quick introduction. And thank you for everyone participating today in this annual VA HAB Task Force meeting. Yes, my name is Lily, and I will be giving you a brief glimpse into part of my research, which is part of my dissertation. And today I will be talking about metagenomic insights into microbial stratification during a harmful algal bloom event in New York River estuary. And so I know we've heard from some of the previous talks from Margie and also from Kim that yes, in 2022, we had a pretty extensive harmful algal bloom in the York River, which was caused by the dinoflagellate Margolipidinium polycricoides, or March. And again, thank you to Savannah Mapes for this great aerial photograph where you can really see the beauty of one of these blooms and that really streaky behavior. And as we've all also already understood from some of the previous talks, is that bloom dynamics are really different, right? So in 2022, which is what this chapter of my dissertation focuses on, we did have a March bloom that never really transitioned into an Alex bloom. In 2018 and 2019, we had no bloom whatsoever in the York River during the summer. In 2020, we had a bloom of March that then transitioned into an Alex bloom. And in 2021, we only had a March bloom that did not transition into an Alex bloom. And whilst much is already known about what nutrient conditions March prefers and what temperature range and what's really necessary for some of these HABs to bloom, we don't really yet fully understand of why every single year there seems to be a different dynamic in these summer harmful algal blooms in the York River. And so one of the components that has been studied but is currently very much understudied, especially in regard to these HAB dynamics, are microbial communities. And so microbial communities, also referred to as microbiomes, are made out of microbes, mostly bacteria and archaea. So really, really small, only what you can see under the microscope, if even. And so these are really important because they're drivers of aquatic ecological processes, namely primary production, organic carbon decomposition, and the nutrient cycle. And so the nutrient cycle that I am really interested in for this chapter of my dissertation, and also what I will be presenting to you today, because of time's sake, is the nitrogen cycle. And so one of the reasons why we are so interested in the nitrogen cycle, as you can probably all imagine, is because March also likes nitrogen, and specifically dissolved inorganic nitrogen forms such as ammonium, nitrate, and nitrite. But so March as an organism is not the only one that is using these forms of nitrogen, right? There's other microbes such as archaea, ammonia oxidizing archaea, or bacteria such as ammonia oxidizing bacteria and nitrite oxidizing bacteria, as well as nitrogen assimilating bacteria such as cyanobacteria, that are all. Share your screen. Share my screen. Okay, that's what I was worried about, that it was not shared. Sorry about that. How do I do this? Unfortunately, I'm not a Zoom wizard. Okay.
Sam, can you stop sharing? Yeah.
Oh, I don't believe I think I'm sharing anymore.
Oh, okay.
Okay, can everyone see it now on Zoom?
You need to switch the camera back to her though. You've got it switched to us.
It looks good on Zoom.
See her too.
That's fine. There you go. Okay, thank you. That's totally fine. I will just continue from this slide now, but just because of time's sake, I basically said everything about this already, but just for the people on Zoom, this is just a little bit of background information of why we're interested in the nitrogen cycle, why we're interested in microbes, and how they're connecting to March, and why that matters for understanding HAB dynamics. So the overall study goal was to evaluate microbial stratification within estuary and harmful algal bloom patches. And again, because of time's sake, I'm not going to go really deeply into my methodology, but in 2022, during that extensive March bloom, we went out on 4 days, during 4 days, and sampled at a location from the surface up to 5-meter depths. And from the water samples that we collected, we carried out a metagenomic analysis, hopefully to answer the following 3 questions. Firstly, how does the nitrification and nitrogen assimilation potential of the microbial community differ between depths? Which microbes dominate nitrification and nitrogen assimilation? And does this information help us understand HAB dynamics in the York River estuary? And so going straight into the results, this is covered now, and I know there is a way to get rid of that, but I'll just read it out. So it says, whole community metabolic potential, specifically focusing on nitrogen assimilation and nitrification. And so the EFPKG value, just to orient yourselves a little bit, is just technically kind of a normalized gene count. And what you can see here is that overall nitrogen assimilation was higher at the surface at 0 meters than at 5-meter depths. However, for ammonia oxidation, so the first step of nitrification, seems to be very similar between different depths. And if we then try and look at the associated prokaryotes for nitrogen assimilation as well as for nitrogen nitrification, what we can see here is that nitrogen assimilation is dominated, I guess not surprisingly, by the Synechococcus, so a cyanobacteria. And we see that there is a higher dominance at the surface than at 5-meter depths. But if we look at nitrification, nitrification was only dominated, no other organism at all, only dominated seemingly from what the genomic data tells us by ammonia oxidizing archaea. And whilst we don't really see a difference here between the depths, which I guess goes hand in hand is what we see with the overall metabolic potential, we do see that there seems to be more of a difference between the days. And since this is just a lightning talk, we're going to go straight into the summary. So what we do see is that whole community metagenomics indicate microbial nitrogen assimilation is higher at the surface at 0 meters than at 5-meter depths, and ammonia oxidation is similar between depths. We do see that a cyanobacteria, the Synechococcus, dominates nitrogen assimilation, especially at the surface, and that ammonia oxidizing archaea dominate ammonia oxidation. And whilst there are similarities among depths, there do seem to be large differences between days from where the samples were taken over the span of this bloom. And so whilst these kind of answer our first 2 research questions that I showed earlier, how does that help us understand HAB dynamics? So what we can potentially conclude from this data is that ammonia oxidation is higher towards the beginning stages of the March bloom, and that Synechococcus may suppress nitrification and may compete with March due to comparable nutrient requirements. And what we're really hoping is overall make use of the metagenomic data to give us an idea of what the metabolic potential of these communities really can be, not just picking out different taxa and looking at them. Whilst this is also what we're doing, we're also interested in what the entire microbial community can do and how it changes because of this HAB dynamics and how these are interlinked. So with that, thank you to everyone. Probably no time to read it all out, but thanks.
Thank you, Lee. We'll try to catch up with questions in a little bit. Right now, we're going to switch over to our next lightning talk speaker, which is Katie Kreider with ODU. Katie, are you able to share your screen? Looks like you're sharing. And go to presentation view.
Yep. Can you guys hear me all right?
Yep, we can hear you. Katie's going to present mapping a Margolefidinium polycricoides bloom transported to the Virginia Beach oceanfront with high resolution multispectral imagery. Take it away.
Sure thing. Yeah, so I'm Katie. I'm a PhD student at ODU in Margie Mulholland's lab. And today I will be talking to you guys about mapping Margolefidinium polycricoides blooms with high-res multispectral imagery. So a little bit of background. I know a lot of you are already familiar with this, but maybe not as much with the freshwater folks. But Marg, as some people call it, or Marge, blooms occur almost annually during the summer months in the lower Chesapeake Bay and tributaries. And these blooms can be very patchy and streaky and are therefore hard to visualize with low spatial resolution imagery. So, some of the work that I've done and Dr. Victoria Hill and some of her scientists in her lab have done is use imagery from planet.com, which has a much better spatial resolution of 3 meters. So that's a 3 by 3 meter pixel size to map the bloom extent and severity. And we're able to do that by using a normalized differential chlorophyll index, or NDCI, which looks at the ratio between the red edge bands of light that the satellite collects. And then you can transform that ratio into chlorophyll concentrations using an algorithm for shallow turbid waters. So for data acquisition, the data was downloaded from planet.com. The imagery is collected by SuperDove CubeSats, which are these really small satellites. And as I said, they have a spatial resolution of 3 meters, and the temporal resolution is about daily, and the imagery is collected around midday. So that's really important for some of these blooms that, vertically migrate, like Margoliphodinium. And the multispectral imagery has 8 bands. And as I said, I was focused on the red and red edge bands for this analysis, but you can also look at the near infrared band of light to mask out land or ignore land during your analysis, because near infrared light is brightly reflected on land, but it's absorbed in the first few millimeters of water bodies. So it's very easy to distinguish land versus water. Or you could use a digital elevation model to ignore pixel sizes that have an elevation of, greater than a certain threshold. So this is the analysis that I completed. I used Esri's ArcGIS Pro software to run the index and then the chlorophyll algorithm. So this image that you're seeing right now is the real color image that you would see. And after running the analysis, I was able to map the total normalized differential chlorophyll index in micrograms per liter. And the values there are kind of arbitrary, but I decided to make 3 classes. But it did a very good job of picking up that streaky, characteristics that we see. Imagery with lower spatial resolution would not have been able to pick up on that quite as well. And this bloom occurred August 28th, 2021. And I specifically targeted that date because there were reports of discolored water. So I, went to that range of dates and started to look for imagery that may have had, bloom features. And there's also been some work in the York and Lafayette rivers by Dr. Victoria Hill and an undergraduate researcher in her lab, Mary. Their workflow is primarily in Python. So I've included a diagram of their workflow here that they made, but it's essentially the same type of analysis that I did, but their final product they would like to create is seasonal mapped products of the bloom presence and the maximum chlorophyll. So they would essentially be able to see which pixels or which locations are experiencing blooms most frequently and which pixels are experiencing the severest blooms or severest chlorophyll numbers. And so they're currently working on this for the York and Lafayette rivers over the 2021 to 2023 period of imagery. This type of imagery, this good 3-meter resolution imagery from planet.com has only been around since about 2020. So there is a limit, and that's a consideration if you're looking at previous years. So this is a slide made by Dr. Hill and Mary. So this is some of their work in the York River. This is just a single day, but they were able to map chlorophyll values into different classes and be able to see, the streaky characteristics of the bloom. And I'm not sure if this is a combination of Margoliphodinium and maybe some Alexandrium too. But I believe, I know my study at the Virginia Beach oceanfront, and theirs is probably primarily Margoliphodinium. All right. So looking to the future, or considerations, if you are interested in using this type of imagery and doing this type of analysis, you can get an educational account for free with planet.com. If you're affiliated with an institution, you can get limited data downloads within 24 hours of the image being collected. And then if you have a government account, you can get unlimited data downloads, but there's a 30-day embargo on the imagery. And I wanted to mention that the planet imagery might not be the best for cyanoHABs because there's some extra bands that have been used for, creating indices and chlorophyll algorithms from the more recent research that I've read. But I'm not quite an expert in that. So, I could look into that more if anyone's interested, if you want to email me. But also Dr. Hill's group is looking into developing an algorithm and parameters specific to the Chesapeake Bay because the current ones in the literature are based on values from Manila Bay in the Philippines. So the community there is going to be different. So they're looking to develop one specifically for Chesapeake Bay phytoplankton communities. And there are some hyperspectral NASA missions going up in the future. So I wanted to put a plug for those here because it's, very exciting products that are going to be coming out, and that data will be freely available. But it doesn't look like it's going to be as great of resolution, both spatially and temporally, depending on the satellite program. But those could be better for cyanoHABs because of the hyperspectral nature of the bands being collected. It's going to be, like every 5 nanometers or so. All right, and so that's all I have today. Please contact me or Dr. Victoria Hill if you're interested in some of these analyses. I'd be happy to answer any questions. This is sort of a side project for me, so I also wanted to put a little research plug of other work I'm doing currently, which is looking at the Marge blooms and their gene expression to determine when they act heterotrophically in the context of other environmental parameters and carbon and nitrogen uptake rates by the community. And I'll be graduating in about a year or so. So if you have any jobs or postdoc opportunities, I would love to hear about them. But yeah, that's all I have for you today. Thank you.
Thank you, Katie. We are going to hold questions. We have 2 more lightning talks to get through. We're a little bit behind schedule, so I'm going to ask Savannah Mapes to come up to the podium and present your talk on advancements with FlowCam and Planktoscope. She's originally from Texas, received her bachelor's in marine biology from Texas A&M Galveston. Before VIM, she studied in the Gulf of Mexico phytoplankton and microalgae for biofuel. She discovered her passion in the harmful phytoplankton and decided to travel to VIMS in the Chesapeake Bay to become a specialist in them. She's advised by Kim Reese and studying the dynamics of York River HABs. Take it away.
Can y'all hear me okay? Works? Okay, great. Hi, everyone. Thank you for coming here today. I'm really excited to talk about this project that some of us in the room and some of us online have been working on for years. We've been talking about it for years. We've been working on it separately, and it's finally coming to full fruition. And the unofficial title that I've given this project is Pixels for Progress, a collaborative endeavor to construct regional image repositories for harmful phytoplankton in the Chesapeake Bay. Now, Kim and Margie already set up the background for this project beautifully earlier today, and so I'll just briefly go through this. In the York River, we experience harmful algal blooms and not so harmful algal blooms throughout the year. And it's several species, but many of these species are prolific throughout the Chesapeake Bay. How do I know that? Well, there are several of us taxonomists, phytoplankton fanatics, sitting at the microscope for hours on end, identifying phytoplankton, counting the cells, and having the best time of our lives. And while this work is enjoyable, it is a bit outdated, and it is time to use modern technology to do this work for us and make HAB sample processing more efficient. One of the ways we're doing that is by incorporating the flow cam. And what is the flow cam? I'm glad you asked. It is a particle analysis instrument that combines flow cytometry, microscopy, and digital imaging into one machine. And altogether, this allows us to use it for a semi-automatic classification and enumeration of cells in a sample. But it also gives us phytofanatics the opportunity for the gratification of seeing the cells and manipulating the cells within the system. Because in order to have this semi-automatic classification and enumeration, we first have to tell the device or the software within the device what to look for. And so this is an example of a species classification library that you build within the system. And I built this for Tripos, Serratium, whatever you want to call it, as the name changes. And so once we build libraries for each of the species that we want to target in the York River or Chesapeake Bay, then we can incorporate them into a digital filter. And this digital filter can be applied to samples that we run through the FlowCam, through either the York River water samples or anywhere else in the Chesapeake Bay. We can run and apply these libraries. And a great thing about these libraries and filters, they can be shared and distributed among FlowCam users. And so if I'm building all these libraries, I can share them with collaborators, and we can all use similar or the same ones and to get similar results. And doing so would streamline the data collection, data processing, and data sharing amongst labs with the FlowCam. And so we've all been working on this separately, some of the labs within the Chesapeake Bay, building these libraries. And so now we're working on sharing the libraries, sharing the data, and coming up with a way that we can streamline the data sharing of the HAB samples. And so we're doing it with the FlowCam right now, but in the future, we also want to incorporate the planktascope. And a planktascope is like a much smaller FlowCam, but it's easier to use. It's less expensive, and it was designed with citizen science in mind. So we can give it to aquaculture facilities in the area, indigenous tribes in the area, whoever wants to have access to see what's in their water samples and protect their protein or aquatic protein sources. And if you're going to be in the planktascope workshop later today, I can talk far more about the planktascope. But that's all I've got for you now. So thank you, and we'll have time for questions later.
Great. Thank you so much, Savannah. The last lightning talk of today is going to be presented by Kayla Marciniak.
Yep.
All right. She is going to present the FlowCam sample collection and monitoring HAB species of the Chesapeake Bay. She is a senior undergraduate student at ODU. I think the other two have died. Her work the past 3 years has consisted of FlowCam operation, taxonomic identification of HAB species, and imaging classification on Ecotaxa, IFCB, and FlowCam dashboards. It should be sharing. Is it sharing? Yep, you're good.
Hello, everyone here in person and on Zoom. My name is Kayla. I know we're also all very excited about lunchtime, so I'm going to wrap us up. I'll be the last one presenting before we go to lunch. As Margaret introduced me, I am a senior undergraduate at ODU in the Mulholland Lab, and today I'll be talking a little bit about the flow cam, its basic function, and how we're using that to monitor HAB species in the bay. First, what is the flow cam? Savannah did a great job of kind of explaining. She took a lot of what I'm going to be talking about today and eloquently presented it. So I will move through quickly, but this is essentially the flow cam and the internal mechanics of it. So on the left, it's just the flow cam with the 2 doors closed. On the right, it's with the 2 doors open. The sample is administered through the top and moves through a series of tubes throughout the machine and then goes to the bottom. And in the middle is where the actual imaging takes place. So this is essentially how the sample collection works. So the flow cell is this area here, and it's a glass tube that the water flows through. The camera and objective is in front of it, and that has magnification just like a microscope does, and that can be adjusted based on the application and the size of particles that you're looking for. And then it has a flashing LED light that is positioned behind the flow cell, and that kind of acts to freeze the flowing water sample in time, and that's how the camera can detect the particles that are in front of it. These particles that are captured are then converted into images, and then images can be put into libraries. So that is how we can classify the particles that we find. So first, the libraries need to be built. So part of my job is building the libraries, creating the filters and the classification templates. A library is essentially a collection of images for a particular particle or type of particle you're looking for. Those can be defined by statistical and value parameters. So that includes things like diameter, biovolume fluorescence, things like that. Libraries are then moved to filters in the classification template. The template is just a collection of libraries that the software can pull from to recognize the different types of particles and differentiate them between each other. This is an example of an image library of heterocapsa. There can be as much difference or like honing in on a particular type of particle that you want to look for. Also, you can create multiple libraries for different orientations of a particle. So from the side, from the top, if there's a particular species that reproduces asexually and you want to see them budding, you can create that in a separate library. So it's more fine-tuned to pick those particles out as it classifies the sample. Part of my job is working on the HAB project with the Mulholland Lab, and my main goal is to run samples through the flow cam to obtain these images and then build the libraries that will later be used for the classification templates for identifying the HAB species. The flow cam is a really important tool because, as Savannah mentioned, it helps kind of streamline the data acquisition process and can take some of the load off of the individuals doing the microscopic evaluations. It can also do cell counts and things like that super quickly, which is super helpful for time management. This is my current list of target HAB species that we're looking for in some images that I have collected with the samples that I have run already. I get a lot of my samples from the VDH GSS shellfish samples, and we have a couple of other places that we sample from, like the Norfolk Yacht Club and Ashland Circled ODU Learning Barge. And one of the other goals of the HAB project is to kind of take all of the imaging instruments, the flow cam, the planktoscope, IFCB, and try to merge them onto a database where we can kind of share all the images together. So we are currently working on a pathway to the flow cam specifically into one of those databases that will kind of help improve identification and monitoring of HAB species and HAB events. And that's all I have.
Thank you, Kayla. That concludes our second lightning round talk sessions. we are past the start of lunchtime, but if everyone doesn't mind, we wanted to try to catch up with a few of the questions that we have. How do I get out of here? Okay, there we go. Okay, so I know we have 5 in the lobby. So from Kathy Wozniak, do you know if you can monitor geosmin from spat, the June Upper Potomac Formidium bloom in 2023 that had high anatoxin was detected after expansive geosmin detection by drinking water facilities?
To my knowledge, I don't think that is something that has been looked at with spats yet. I don't think we've really gotten into like taste and like odor compounds yet. So to my knowledge, I don't think so. I probably not like the expert to talk about that. But from everything that I've seen, I looked this up a little while ago now.
Oh no.
Julia,
Okay. Yeah, there we go. Okay, so Paul, you want to help me out with this one? I would assume that it could be absorbed to the spat since it does have non-polar entities, which are leading to us being able to smell and taste it. Would you kind of agree that it probably would affiliate? But I haven't seen any papers. Have you, Paul? Calling out Paul Zimba here. Any thoughts? Oh, no. Let's do this so she can hear. Pass.
Hey, Julia. I've never actually had geosmin or MIB detected on any spats, and I would have recognized it if I smelled it. So I don't think that it is well retained by the spats.
But there might be other resins that could work, but haven't been tested, just to kind of, it's still a doc, just not with the same resin.
So Juliet said there might be other resins that could detect it.
Well, okay, but that brings up a very good point. You know, the specificity of spats is a huge.
Sorry.
There's pretty good evidence that that not all toxins are the same. You know, in the
Resins are the same.
excuse me.
And not all resins are the same. Yeah.
And Juliet should be the one talking about this. There are at least 3 choices, though, of polar, non-polar absorbents that I think are good alternatives to consider.
Okay, there was a June Upper Potomac Formidium bloom in 2023 that had a high anatoxin was detected after by the drinking water facilities. Was that concentration in mats not dissolved? Is that a question or a statement, Kathy? Statement, I guess. That concentration was in mats, not dissolved. I think that's a statement. Somebody asked, thank you for the presentation, Samantha. I think you said that the spat bags had beads in them. Oh, do you know what the beads are made of, natural fiber or plastic? Okay, so can you tell them what the beads are made of, Samantha?
Yeah, to my knowledge, I know that it's a non-polar like copolymer type resin. Beyond that, I don't know if I can tell you too much. I just know it's an adsorbent resin, and it is non-polar. But that's about where my knowledge with that starts and stops. I can give you in the chat the exact specific type of resin that I use. I think it's from Sigma Aldrich, I want to say. But yes, I can send more information. I might need to look it up a little bit, but that's kind of as much as I know.
If it's the diazon HV20,
Yeah, I think. I
It's
just wanna
a
snap
it's a copolymer mixture of divinyl benzene and polystyrene.
Yes, that is exactly what I use. Thank you so much.
Yes, but I'm gonna assume that that's the resin. Okay, have you or others, oh, okay, I think this is for Samantha also. Have you or others done or published the uptake elimination curves or rates in the lab for the spats for these toxins? Just getting at equilibrium-based sampling for passive sampling devices. Oh, yeah.
So no, this is not something that I have done myself. Again, this was just kind of a general pilot study this year. So it was essentially just kind of seeing how I could deploy my spats and then just kind of trial and erroring a lot of my extraction methods and everything like that. So beyond that, I haven't been able to do that much more work. As for publications, I'm not entirely sure. I know a lot of other people have worked with spats here in the task force meeting. I don't know if anybody might be able to add on to that question.
You want to go?
Okay.
I can give a quick comment that for most of the toxins that were presented in Samantha's talk, I don't know that the uptake, so absorption and desorption rates have been calculated in a controlled laboratory setting or in the field per se. But some of the other toxins, like the marine lipophilic toxins, those rates have been calculated. And just recently, I think at the last HAB symposium, Rafe Kudella's lab is about to put out some information they presented at the HAB symposium, where they were looking at uptake rates of a few different lipophilics, also domoic acid and microcystins. And it looks like microcystins and domoic acid specifically do come back off the spat. So that's something that everyone's starting to pay attention to now. And that was one of the ideas of the SIPS membrane, is that since it's infused with toxins, you can look at loss of toxin, the ones that Josh had presented. You can look at loss of chemicals from the resin while you're deploying, so that now you can start to make some calculations as to loss over time while you're also absorption, so that you can get into things like, have you reached equilibrium? Have you reached capacity? Are you still in the linear uptake phase, or are you starting to reach that state where you're no longer binding toxins actively? So I would say there's definitely stuff to look in the literature. I would look for some stuff coming off of Kudella's lab in the next couple of years. I think that'll be relevant to the freshwater stuff.
Okay, so you guys in the room can see this, but Natalie Hall's developing a HAB spectral reference database using the microscope-based hyperspectral system. If anyone likes to collaborate and send samples, please contact her at nhall@usgs.gov. Freshwater samples primarily, but estuarine as well. And it can feed into the orbital hyperspectral emission data collections.
Do we have any questions in the room for any of the prior speakers this morning? I think we got hungry people.
Yeah.
All right, Kim, can you wrap us up for lunch with the folks online?
Okay. So everybody, we're going to be coming back. We'll come back right at noon. So unfortunately, our lunch break, at 1, I'm sorry. Yeah, we're going to be back at noon. We're going to, space warp here, time warp here. We'll come back at 1. So for those of you online, we'll have like a 44-minute break here. The lunch, I think, has been set up in the atrium here. And then, as I said, you can go into rooms A, B. They prefer you not eat in here. Okay, so rooms A, B, C. We might be able to get into the director's conference room now, which is also down and around the corner. I don't know what the temperature is outside, but somebody might want to go. It's freezing in here. I know. I am shivering. I don't know about you guys, but meanwhile, let's try to get the camera back here. So please help yourself. There are drinks and coolers out there. There's a lot of food. And there should be chips, cookies, sandwiches.
She was here, but they married to, they think.
The General Assembly House Bill 30 HAB investigations. To provide a quick overview, we have Drew Gary with the Department of Environmental Quality, Water Quality Monitoring Team Leader at Virginia DEQ, coordinates agency field activities associated with HABs and activities of DEQ with HAB task force partners. Put this to your follower.
Margaret, I'm not sure we could hear you. What did you- Oh. I thought you were-
She said, I'm Drew.
Oh, okay, got it.
Here for 2 minutes. Yeah, if I have 5, Oh, good afternoon, folks. So these are the House Bill 30 harmful algal bloom investigations. It was HB 30, and I think this 2022 session, HB 30 now would contain.
Sharing your screen.
Am I sharing my screen? I don't know. I am not.
I like the term Zoom wizard.
Thank you, Todd.
Thanks, Todd.
Yep. What do I gotta do to become a Zoom wizard?
So these are the House Bill 30, General Assembly Session 2022 algal bloom investigations, which I'll explain here. So due to numerous no swim advisories on Lake Anna in recent years, I think they really upticked in 2017 or 2018. And then benthic bloom reports in the Shenandoah River, 2021 on the North Fork, there was this big advisory. And then 2022 to present, fairly regular occurrence of benthic blooms and occasional benthic alerts, awareness alerts from VDH. There's this 2020 allocation from the General Assembly. So in 2021, the General Assembly ordered us to convene DEQ to convene this multi-agency work group to write a report on HABs. And they basically wanted us to describe the extent, magnitude of HABs, where they were bad, where they weren't, and how we were going to solve it. And we effectively got less than a year and $0. So we wrote a report and said, here's the extent to which we know, and here's what we're doing and not doing. And a lot of more information is needed, especially system-specific intensive studies are needed. So in 2022, they allocated $3.5 million to us for Lake Anna and Shenandoah River HAB investigations. And I was going to put the language up here, but it's rather open-ended. It says, study the algae in these systems, basically, and go. No identification of what goes where, but we sort of figured that out, how much to allocate to each system. We're the lead agency, but we coordinate a lot of groups. I hope I mentioned everybody here. This is definitely a multi-group effort. BDH serves as a very key advisory partner in this whole these projects. ICPRB and USGS, they're the lead study partners on this. They're the ones that are going to be talking about the research today. Key study partners also, Old Dominion University, George Mason, VCU, VIM's also helping out in an advisory capacity. So this is really a multi-group partnership. Field investigations, 2023 and upcoming in 2024. That's the data collection phase. And then a report will be out in 2025. And big and broad, really, we want to determine the characteristics, frequency, and magnitude of these blooms. What are potential causative factors, natural and pollution related, and what are potential predictors? And then if we can, develop some strategies for prevention and mitigation. So with that, I will turn it over to our partners who can tell you about the important stuff. Thanks.
Thank you, Drew. We have Carly Moss with the USGS. She's a hydrologist at the USGS West Virginia Water Science Center. She graduated from the University of Maryland in 2022, the master's in geology, and joined the USGS in 2023. Thank you.
Right. Hi, everyone. I'm Carly Moss, and I'll be presenting on the drivers of harmful algal blooms in Lake Anna, Virginia. And Drew did a really great introduction, so I'll be going straight into the project objectives, give a brief overview of Lake Anna, discuss our study design and our preliminary data. We began studying in Lake Anna last summer, so this is very preliminary data, but wanted to share with you all what we're collecting so far, and then touch on our next steps. We have three main project objectives. The first is to identify and understand different factors or different processes that are causing HABs to form, persist, and decline in the lake. Then two, identify primary sources of these factors that are contributing to HABs formation. And our last goal is to identify different technologies or unique approaches to help predict or provide early detection of harmful algal blooms. We'll go over the study area. We're studying in the upper Lake Anna watershed, and we have 6 sites that have been established for this project. I was trying to see if I could get rid of that bar. Anyway, we have 2 sites in the tributaries, one on the North Anna River and one on the Pamunkey Creek. And these 2 tributaries feed into the 2 main arms of the lake. So this arm of the lake is the North Anna arm, and on the opposing side is the Pamunkey Creek arm. We have another lake site, which is downstream of the 2 arms where they come together. And this is our sites. The site will be called the split site. Finally, we have a meteorological station at Belmont Park. It's taking a second to progress. There we go. The land use in the Lake Anna watershed is pretty patchwork, which is typical of Central Virginia. The dark green in this map is forested areas, and the 2 shades of light yellow are agricultural areas. The land use at the Pamunkey Creek watershed is about 58% forested and 31% agricultural. The North Anna River watershed is more forested and less agricultural. And the watershed as a whole is about 65% forested and 21% agricultural. Next, I'll discuss the data. What instruments do we have out at the lake sites and the tributary sites, and show some of the preliminary data that we're collecting. First, I really wanted to give a huge thanks to the team. They've been really dedicated scientists and have had to learn new techniques to work in a lake environment. They've not only had to be very dedicated scientists, but also learn how to code, be electricians and carpenters. Wanted to give a thank you to our dock owners for letting us always come out on the lake whenever we need to access our equipment or to sample. And finally, LACA for support throughout this entire project. Our sampling plan is in 3 different parts. We have a meteorological station. We have our tributary sites and lake sites. And I'll go into these in more detail now. First, I'll start with our meteorological sites. We have a full meteorological station at Belmont Park. Here, we're collecting 5-minute measurements of photosynthetically active radiation, or PAR, and this measures the usable light for photosynthesis. We also collect precipitation, air humidity, temperature, wind direction, and wind speed. And we have another wind sensor on the North Anna to get a better picture of what's happening at the lake. Temperature, rain, and light are known influences of HABs. And so I wanted to show what we were seeing in Lake Anna watershed. The temperatures are very typical of Central Virginia, with a range of temperatures from -8 degrees C to 37 degrees C. We've had 23 inches of rain since we installed our meteorological station in July, and we're looking forward to parsing through this data to understand periods, dry periods and wet periods, and look at rainfall intensity and duration. And we have our PAR sensor here as well. This is a graph over time from June of last year to mid-January. Each of these blue dots represents a measurement that we collected. So during the summer, when we have long, sunny days and really direct sunlight, the PAR is higher than during the winter when, our days are a lot shorter and the sunlight is less direct. And this is following in sequence with our daylight hours. Next, I'll discuss the continuous and our discrete efforts at the tributary sites. We have 2 monitoring stations, one at the North Anna near Monrovia and one at the Pamunkey Creek near Lahore. And these are just images from our sites. Here we have water quality measurements that are collected every 15 minutes with water temperature, specific conductance, pH, dissolved oxygen, turbidity, and a nitrate sensor. And we're also collecting 15-minute measurements of gauge height. And these are all available on the USGS's websites for these 2 locations. We found over the past few months, so this is data from August to mid-January, that the 2 tributaries are very responsive with precipitation, which is shown on this graph right here. During these precipitation events, we also have seen that the nitrate concentration, particularly at Pamunkey Creek, which is the more forested watershed, really increases during these precipitation events. And we're looking forward to seeing how this looks over the course of a year. But the North Anna River in red has not been as responsive as Pamunkey Creek. So during storms, we've observed increased gauge height and discharge and potential nitrate runoff from different watershed sources that we're looking forward to investigating in the future. We also go out and collect water quality measurements as well as discharge to get a better understanding of the loadings into the lake. We go out once a month and we collect samples at the 2 tributaries, and we also go out during storm samples. We're able to capture a lot of the storms that happened in December and January. We have depth and width integrated samplers and use isokinetic samplers. The different water quality constituents that we are measuring are major ions, nutrients and sediments, stable isotopes, and photosynthetic pigments. Here's a snapshot of some discrete data that we've collected at the 2 tributaries. We've been able to collect some storm samples here, but we don't have data back from the lab past November. But here are graphs of nitrate over both time and with increasing discharge. Over time, the North Anna and Pamunkey Creek have very similar patterns and go kind of hand in hand together. And there's increasing concentrations with increasing discharge. Orthophosphate has a similar pattern to nitrate where the 2 tributaries follow closely together, and there's increasing concentration with increasing discharge. I also wanted to show chlorophyll because I found that interesting. At the Pamunkey Creek, there was always higher concentrations of chlorophyll in comparison to North Anna, and there was still, in general, increasing concentrations with increasing discharge. But we're really looking forward to getting back some data from the lab and seeing this, how the storm samples fall in line with this data here. Next, I'll talk about our lake sites where we have continuous, discrete, and special studies efforts on the lake. Like I mentioned earlier, we have 3 sites on the lake, one on the North Anna arm, one on the Pamunkey Creek arm, and then one downstream of the confluence called the Splits. Here's an example of a dock box that we have on our homeowners' docks that holds all of our equipment and allows us to transmit the data in real time. Here we have 3 different instruments. We have a water quality instrument that takes 15-minute measurements of the big 5, water temperature, specific conductance, pH, dissolved oxygen, and turbidity. We have a really cool instrument called the phycoprobe that measures chlorophyll and unbound phycocyanin in 15-minute measurements. And from this instrument, we're able to collect total chlorophyll data, green algae concentration, blue-green algae concentration, diatoms, and you can read the rest of the list. And finally, we have a wave action sensor, which is collecting quarter-second measurements of wave height. So then we can better understand maybe when waves are coming from, whether they're coming from boats that are driving by or from wind, when these waves are occurring, and how high the waves are. wanted to show some data that we're collecting from the 3 sites, and this is data that's available on our USGS websites as well. Here is our graphs of pH and dissolved oxygen with the same scale on all 3 graphs to show dial changes over the day and also seasonal changes. So first off, at Pamunkey Creek Arm, there seems to be really drastic changes in pH and DO throughout the, particularly in the summer. The maximum DO range over the course of a day at the Pamunkey Creek Arm was 8.4 milligrams per liter. And North Anna has even more drastic changes, where the maximum DO range in a day at that site was 12.4 milligrams per liter, with the DO range being greater than 10 milligrams per liter over 11 days. However, as we moved our way downstream to the split site by this confluence here, the DO range, the maximum DO range in a day was only 3.1 milligrams per liter. So we found that there is greater dial cycles at sites closest to the tributaries, and these dial cycles can be indicative of photosynthetic processes that are controlled by algal growth. And greatest change was at the North Anna Arm. I also wanted to show some data that we're collecting from the FICO probe that can measure the chlorophyll and the unbound phycocyanin in real time. This is just a snapshot of about 30 or 60 days, so very, very preliminary data. But we have green algae on the y-axis with the Pamunkey Creek site in green here, the North Anna Arm in red, and the split site, which is most downstream, in blue. So just from this snapshot of data, there's the higher concentrations of green algae closer to the tributary site, so these 2 lines. There's also these declines in concentration that seem to be very rapid. And so far, these changes are co-occurring with precipitation events, but we're looking forward to having more data and looking at this in a different way or over time with all the instruments that we're measuring. We also collect discrete data at the lake sites. We go out twice a month during the growing season and once a month during the non-growing season to capture the annual changes. We collect our samples using a camera, which is seen here. If the lake is not stratified at our sampling sites off the dock, then we collect a composite sample. But if the lake is stratified, we collect 2 samples and treat them as if they're 2 completely separate samples. Here we have water quality constituents and algal constituents. The water quality constituents we're measuring include major ions, metals and trace elements, nutrients and sediment, biomass, and Secchi disk depth. And the algal constituents that we're looking at are cyanotoxins, photosynthetic pigments, algal community, and we're analyzing for environmental DNA and environmental RNA to understand if genes are being expressed at Lake Anna. ODU is doing our analysis on the phytoplankton community composition and toxin concentrations. We've had really good presentations on the flow cam, so thank you, Savannah and Kayla. And we're also measuring toxin concentrations, which I'm sure you can talk to Margie about and get a really detailed understanding of what they're looking for in measuring. Here, I know these are a lot of graphs, but I wanted to show you that this is just some of the data that we're measuring at Lake Anna at the 3 sites. And here I'm showing phosphorus, nitrogen, and carbon. And I just wanted to give our takeaways so far. We've seen the highest concentrations at the North Anna Arm, which is the site right here, in particular in forms of phosphorus, total nitrogen, and carbon. And we've seen patterns of decline in nutrient concentrations after September. But so far, we only have data from June to November back from the lab, so still very preliminary. We have a suite of different special studies that are going on at the lake so that we can better understand temporal and spatial variation at the lake. And I'll go through all those here. We have a field parameter synoptic where we get on a boat and we sample at all the 30 sites at these red dots throughout our study watershed in the upper Lake Anna. And here we're looking for changes in water quality and algal parameters across the lake and with depth. We conduct this once a season. At each of these 30 sites, we also are assessing the amounts of nutrients in the bed sediment, and we went out and we collected this data last August. We're collecting for nearshore transects at 3 sites, these yellow sites that all have different land uses, to understand the impacts of how nearshore activities like mowing the lawn, applying fertilizer, or septic systems are impacting algal community and growth, as well as nutrients during periods of low residency and during periods of high residency. Our last 2 studies are conducted at our USGS lake monitoring sites in the bright green. Our wave action study will monitor the impacts of waves on different nutrients in the water, and we'll go out and collect samples during a low wave day and a high wave day, like around a holiday. And our last study will be to model lake metabolism at these 3 locations, which will hopefully be used to provide early indication of harmful algal blooms at Lake Anna. Next, going into the plan. So we've started addressing the problem, looked at our project objectives, and over the past 6 or 8 months installed and began intensive and extensive data collection. And in the upcoming time, we'll continue and finish our data collection, analyze and interpret the data, and begin our publications. Thank you for listening, and I'm looking forward to answering any of your questions.
Thank you, Carly. So Carly presented on the Lake Anna portion of the studies. Are there any questions online for Carly?
Not yet.
Any questions in the room?
Yeah, you have 2 sites where you're checking rainfall.
Just one.
Oh, just one.
Just one at the Belmont Park there.
I know at Smith Mountain, we run into the rains coming through and it's very sporadic. And with your big watershed like we have, you know, do you feel like you're getting enough flow information to cover the area? I mean, you can't put a weather station everywhere.
Yeah.
But it's hard. I mean, that's one of our big concerns is, you know, in Cocoa Roz, the citizen science thing, I've tried to use that. We've used weather stations at the airport. You know, you try other things, but with one site, it's kind of hard to draw correlations if the water's flowing in or if a front moves through and it's sporadic rainfall. Do you have any ideas about what you're going to do about that? Or are you just going to hope for the best?
I think right now we have a lot of information coming in at the meteorological site, and so I think we're going to take that data. And our watershed is a lot smaller than at Smith Mountain Lake if you're trying to cover the entire lake. I think we're gonna roll with it.
Well, and you have to.
Yeah.
You know, you don't have much choice, but it's the big issues.
Absolutely.
So sporadic and in one part of the lake. We tried to put gauges out for the monitors, the volunteer monitors, and you know, just to get rainfall.
Yeah.
Turned out to be so different.
Interesting. Yeah, that's good to know. But I think our watershed's a lot smaller, so I think we'll be okay.
Hi, Carly. Thanks for the rundown. And just to this point here, I understand there's a big network of volunteer weather people that's been going on for decades, and that might be another source. Also, the watershed, I think, goes up towards Charlottesville at the very headwaters, so you might triangulating several weather stations. But I was, I'm sure you're planning on doing this, hopefully, but it'd be really nice to see the correlation between the FICO probe data for different groups and the actual taxa collection. So I'm looking forward to seeing that.
Absolutely, that is the plan. Yeah.
Has the probe been out?
Since mid-November. So
You had fouling or any problems with the probe itself?
It definitely gets dirty really quickly. We have it in a flow through cell, and so we have had to go out and clean it. But the data is still preliminary, and we're still learning how to use the equipment.
Yeah,
But
we
so far it's
had a
working well.
probe out and just siltation when the boats are going by and stuff was a big problem with ours. So
Was it in the water?
yeah.
Ours is in a flow through cell, so we have a pump that's pumping it into
So
our
that's
dock
helpful.
box.
Oh.
Yeah, so I think that's
I'd
definitely been
like
really
to talk
helpful.
to you about that. That sounds like an interesting approach.
Yeah, absolutely. Let's chat later.
Any other questions in here? Questions online? Okay. Thanks, Carly.
Thank you.
All right, so we've gotten the update for Lake Anna. We're now going to get the update on those studies for the Shenandoah River. And for that portion, we have Gordon Mike Felkman with the Interstate Commission on the Potomac River Basin, or the ICPRB. And he's just going to bring up his presentation and get started. Got it.
All right, can everybody hear me? All right. Slideshow from beginning. All right, well, I guess good afternoon, everybody. Sorry to catch you after lunch. If I catch anybody sleeping, I'm throwing something at you. So this project, today, what I'm going to present isn't so much data as much as it is the organization, the logistics of the project, what we're trying to accomplish, and the roles of the different partners. Truth be told, we have some preliminary data, but we're just getting to looking at a lot of it. But what I'm hoping you leave this presentation with is that the intended coordination and the products that come out of these different pieces appear to be working, and everybody's getting along well. And it's a pretty exciting project that we have so many irons in the fire, and they all seem to be cooperating. So moving forward, this is a lot of language that Drew already talked about. We have the link here on kind of the history of the project, so we're going to simplify it. Really, the project objectives kind of consolidate down to, we want to inform the extent and the risk of benthic algae blooms. This is different than the pelagic blooms that we've kind of talked about in estuarine waters and in Lake Anna. This project is really focused much more on benthic attached blooms. That's an important little nugget. Number two, again, we're interested in identifying the factors and the processes leading to the HAB formation, persistence, and decline. The reason I've highlighted these handful of terms is that really to start, this is a pretty new project. It all consolidates down to what is it, where is it, and when is it there? Going to the third piece, we want to look at things like water quality and see if we can get some of these predictive variables and kind of understand the story better. But really to start right now, those three questions off to the left are kind of what we need to nail down. And then finally, this fourth objective is to identify technologies. So as I go through this presentation, I'm going to kind of point at some new tools that have been brought into the Shenandoah that weren't there prior. And I think are really kind of pushing the ball down the field as it goes. So the coordinated effort, the way that this presentation is broken down is there will be a column for coordinated field effort. There will be the labs that are working on it. And then off on the far side, which will be next year, is basically the consolidation analysis phase and interpretation. So this coordinated field effort has 4 major partners. Truth be told, there should be a 1E because when we started this project, GMU was kind of the lab that we were going to send a lot of this to, but they've taken a very active presence in the field effort. So there is kind of a hidden, we'll put 1E just below the presentation screen. So USGS is responsible for continuous data and water column samples. ICPRB is going out in the field and actually taking these benthic algal samples, both qualitative and quantitative. I'll get into that in a little bit. Virginia DEQ is working with USGS and taking water chemistry along the entire survey reach. And VCU is bringing some really exciting new technology, some drone imagery to complement what we're doing out in the field. So all these parts have to agree. And again, this presentation is just thick with links and people to contact because I don't think it's possible that I could get into the weeds of every part of this. So jumping in, we have two rivers that we're really looking at. There's the North Fork Shenandoah and the South Fork Shenandoah. Because of logistics and just the reality of personnel and funds and things like that, we put a lot of effort into the North Fork Shenandoah. This is a bracketed approach. So by cooperating with USGS, we have our upstream research point, which is down in the bottom left of the map. That's Mount Jackson. There's a USGS gauge link there. Going all the way down to Strasburg, which is just upstream of the confluence at Front Royal. So we're covering the affected area of the 56.5-mile bloom, the closure that I think brought a lot of attention to this. As we're going through and doing this sampling, there's USGS working on the ends. We know what comes in, what goes out. The points in between are ICPRB going out and collecting benthic samples and delivering those to GMU because, again, logistics would be a lot for a single field crew. We have DEQ and their field crew going out. In any of the places where you see a square, that is where DEQ is taking paired water quality samples with the benthic samples. So when all this is working properly, there's the non-tidal network trigger. USGS is going out, letting us know when they're going out, and then ICPRB and DEQ are following right behind. So all this is done within a 48-hour window. So it's as good as we can get of a fully described kind of grad. The longitudinal survey sites were selected based on accessibility, semi-wadeable. This has been a kind of a laughable term. North Fork's great. It's nice and shallow, especially in a drought year. When you're my height, it's great. When we get to the South Fork, even I complain that it's a little bit deep. And this year, we're probably going to bring masks out to take samples. We can wade out there, but any sort of rain event, that pushes this out. I'm kind of jumping to the next slide, but so yeah, semi-wadeable historical bloom location. Again, we've heard about that a lot today. And we wanted to try and hit points as equidistant as possible within the brackets. Unfortunately, two of the sites that were originally selected on a map, once we started going to them, depth and things like that just weren't going to allow. So we've relocated those sites to be better described by drone flights down by Strasburg. It's closer to the Strasburg or the Strasburg USGS location. So although it was intended well that things were going to be nice and spaced out, the reality of the river just wouldn't have it. So yep, and then DEQ with USGS, there are 5 points along the North Fork where we have water quality with the benthic samples. The South Fork doesn't get the same level of technology. I'll get into some of that technology in a little bit. We're out there, we're taking benthic samples, we're trying to understand what's going on in the system, but most of the effort right now is focused on the North Fork. Again, we have 5 points that DEQ and USGS are getting our water quality data. It's all the same trigger. This is all happening within a 2-day window. So what is USGS doing? They have continuous monitoring stations, discrete grabs, and synoptic sampling. I'm not going to spend too much time on this, but you know, there's a lot of really good data coming out of these 15-minute continuous data sets. Some of the discharge data goes back 100 years. We have solar radiation every 15 minutes, water quality with the YSI, the EXO, water temperature, conductivity, pH, DO, turbidity. One of the new pieces of technology that was introduced to the North Fork is the nitrates sampler. Pretty exciting to see that high resolution data delivering, and I'll show some of those slides coming up. And then we also have a chlorophyll, the FICA probe that Charlie had mentioned in the Lake Anna study as well. The USGS is bringing a lot of technology, a lot of really powerful, both long-term and high-resolution data sets to this project. Not going into all the pieces of it yet. Like I said, we haven't really had time to interpret a lot of the data, but we have, 2023 was a drought year throughout the Potomac, particularly parts of Central Virginia. And you see it kind of break there at the end of November. The nitrate data, my God, the sensors are working with the price they are. That's a happy thing to see. Still need to take a look at what kind of trends are happening with nitrate, but again, it seems linked with some of the flow spikes that we're seeing. The continuous DO and pH goes back to biological metabolism. Again, things that were kind of discussed with the Lake Anna project, but really exciting to see these at Mount Jackson and Strasburg, because that pH of 9.0 is kind of one of those magic numbers that when you start looking at nutrient availability and changes in chemistry, you start seeing this potential for like positive feedback loops in chemistry and nutrient availability shifting in the environment. So really good data to have out there. This is just a great picture. And again, thank you, Brennan, for putting all this together. This is a live feed camera that we can see what's going on at the upstream site. And a lot of application to this, between everything from flow to understanding when is a good time window to even look at taking a picture of algae growth and stuff like that. So a nice link there. And then we're moving to the discrete data. Again, the sampling is once a month, and then there's also storm samples being collected. A lot of the same suite as what was described with Lake Anna. The thing that I'd like to point out is that nutrients, nitrogen, phosphorus, and carbon in both total and dissolved forms, and then the major ions. Some of the things that we've talked about in previous Virginia HAB conferences is, again, this idea of non-nutrient chemistry and how the biology of the organisms with the kind of unique ionic chemistry at these sites, how it all plays a role in these production. What's also happening at these sites, USGS is sending off DNA and RNA assays to their Ohio lab. So that is another kind of supporting data set. Finally, getting to the synoptic collections USGS is doing. Really excited about the groundwater work. For those of you familiar with the geology of the Shenandoah, there's a lot of groundwater. And it can show up as a temperature anomaly. It could be in stream. You can see a cool spot from a bank leading out. And then finally, they're taking some of the continuous dissolved oxygen and pH work and looking at stream metabolism. So moving from the brackets, the two ends of the sections, to our role, ICPRB's role, and looking at the benthic algae. So at this point, everything USGS has done has been primarily water column samples. We're focusing on the benthic samples. There are quantitative and qualitative samples being collected. There are the 10 sites that I've already mentioned, but there's also this thing that we're calling the response bank. That is taking canoes out and actually trying to track down blooms between those regularly visited sites and understanding what's happening at sites that would otherwise go unreported. At each one of these sites, there's a quantitative algal sample, which was based off of, if you look off to the far right, California's swamp method. There's a link there where they actually use this as an IBI, a measure of biotic integrity, and a whole suite of other things. And algae was just one of these components. They used 11 transects that would go out, and there would be a YSI kind of water quality suite taken at each point, along with algae samples. Truth be told, this is a great method. It is time consuming. The first time we did this, it took us 4 hours to get one site done. So we had to modify California's swamp method for Virginia waters. We can do 9 transects with the same randomized start. We can still take quantitative and qualitative grabs. The YSI profile is at the top and bottom of each site. and the supporting water quality, like the nutrient chemistry and things like that, are being collected by DEQ and USGS. So we were able to kind of reduce the labor of getting these samples. Rapid fire, this is just a standard delimiter. And with that, we're able to cut out, remove the algae that falls underneath the selected 9 transects, the subsample sites. In the California method, wherever that point landed, you collect it on. What we found in the Shenandoah, particularly when we looked at the composition of what was growing out there, we weren't collecting on wood or macrophytes as best we can. And predominantly, just because of the way the Shenandoah is, we were sampling on finer sediment, cobble, large gravel, small boulders. But that scrubber figure 8 there that came from California, it doesn't really play well with Shenandoah bedrock. So we're trying to improve that method as best we can. Once those samples are collected, they go on ice, not frozen. They're kept out of sunlight. And through cooperation with GMU, a lot of times Chris back there drives out and will actually pick up the samples on day 1, and then we drive down to GMU on day 2. So there's a lot of driving time, a lot of podcasts being covered. Qualitative samples. This is if we see a bloom, but it doesn't fall at one of the random spots that we're going out and collecting from. The qualitative samples are basically going out and targeting, wanting to get a sample that represents what was at that site, even if it was outside of our regular grabs. This has kind of turned into a project on its own, as it's helping us ID what's out there. Our in-field images are being paired with one of the professors at GMU's macroscopic photos, and we're building an image library to help train people on what's out in this river. And from these qualitative grabs, this is the first kind of, I was super excited when I saw this table come together. Those first couple questions of what's there, where was it, and when. If you look, we have our taxa off on the left. We have the dates along the top column of when they were observed, as well as the sites, which unfortunately the top bar of Zoom looks like it's taken from you. So we start having an idea of kind of what was described in some of the estuarine data earlier today, looking at the succession of species over the course of the season. So this is pretty exciting to see. Finally, VCU and their aerial imagery. There's Paul Zimba and Ron Lopez there. They have brought a $250,000 to $300,000 drone into the new technology in this project. This is a hyperspectral camera attached to that drone, and it's producing images like this, with the left being our ortho image and our time series of a particular bloom site off on the right. And just a weakness of the study, using 9 sites to represent, let's say that this section of river is 20 meters wide, 50 meters or 250 meters long, 5,000 square meters, just back of the envelope. Those qualitative grabs or quantitative grabs that we're doing, they're covering like 0.1% of the total area. So it really statistically, it's not serving the role of total coverage. It's serving the role of what's there and getting at questions like biomass and building a pigment library and things. This is our best attempt so far at getting a coverage estimate. I'm pretty excited by these results. So moving through, these sites are where normally we're working upstream of bridges and like road crossings. VCU, just because of logistics, they can get 2 flights in if they're at a single point, so they're going upstream and downstream. The downstream sites are outside of the kind of focused area of the other grab samples, but we're still getting the paired water quality and things like that with it. So by kind of doing an upstream and downstream, or in this case, west and east approach, we have 3 really well-described sites using this drone technology. Just moving through, VCU is ground truthing their samples to look at their pigment analysis. analysis and to start getting estimates of total coverage to differentiate between things like chlorophyll A and filamentous green algae and then the cyanobacteria. So there's a lot of promise there. It's a function of resolution, as I understand it. But there's a lot of pre-processing, a lot of post-processing of the imagery. So really, the image lab is another lab that should be highlighted here as well. Moving on, participating labs. It's almost like that segue was supposed to be there. GMU is really the focus of a lot of the algal samples and what we're collecting in the field. That is Dr. Chris Jones and Dr. Rosalina Krisnova. And they are receiving our algae samples and doing taxonomic IDs, pigment analysis, toxin analysis, and biomass. This is the super fast rundown. And for those of you that were here in the morning, one of the students had already kind of presented on the algae sausage. So with this one, all of our algal samples are delivered within 30 hours of collection, again, held just above freezing, but they are homogenized. In the bottom right, you see that the sample is dumped out, basically spun around to make it as mixed up and random as possible. Large fragments of detritus, stones, invertebrates, things like that are removed. And then from that flat cylinder, you end up with a quantitative breakdown. So if the cylinder is the title quantitative composite cylinder, you see that it's cut into 8 sections. There are 2 ELISA, 2 LCMS, dedicated pigment process, ash-free dry weight, which is getting at our biomass measurement, a taxonomic macroscopic ID, and then off to the far right, I called it when we started this project, it was the kind of orphaned piece of sausage. We didn't know what was going to happen with that, but we knew it could be stored and it would be looked at. And I'm excited to say that Chris has told me that GMU is actually looking at molecular analysis of that last piece. So we're getting a full suite of each one of those samples, all related with paired water quality, as well as having the continuous data upstream and downstream. It's a pretty complete package when it's all said and done. So what's happening with the year ahead? USGS is going to continue operating the gauge and the Kanban stations. We're still using the NTN, the non-tidal network dates as our triggers to go out, weather and the world providing. Year 2 of this multi-agency coordination. If last year was difficult, this year is going to be more difficult. There's a lot of communication that goes on, but we want to try and, by moving one of our sites down to Strasbourg, we want to try and further incorporate the drone flyovers. The 10 sites that I mentioned that were the static sites, they're going to be supported by longitudinals this year. Like I said, there are 12 events in the bank that we're going out and responding to these blooms. So there's going to be more samples being delivered to GMU, as well as coordinating with DEQ and the partners on this project of what we're seeing out there. Nice thing is, so we started our first date out last year was at the end of July. So our first good full collection was in August. At that time, basically having the supplies, things like the pigment standards and staff to even run and process these samples. At the time, Christina wasn't even with GMU. She was on her way. So we have a full taxonomist, freshwater taxonomist at GMU now. But we're happy to say that GMU is now fully staffed. There's an entire undergrad army, it sounds like, that's excited by this project. And there's a lot of tangential and supporting things being done. VCU is going to continue their hyperspectral drone flights and ground truthing. And again, I think there's going to be more discussions this year about cooperation between the partners. And again, we're going to opportunities that aren't really part of the promise package, but they're sort of developing on their own. We're going to refine the sampling of sort of a regional field guide. The spat sampling, I'm very excited about because right now grab samples with toxins are kind of hit or miss. So a passive approach might be something that sort of strengthens that argument. And then there's it was presented earlier today, but the gold standard strip test, something that can help us rapidly assess whether or not, toxins are over a certain threshold. But I guess in sort of wrapping everything up, the science that USGS has been able to bring along, DEQ with their field crews and their assessment branch that really have helped out a lot. George Mason with their lab, VCU with all the drone work that they've done, and ICPRB with trying to get the samples together and stuff. It's just I'm heartened by the amount of collaboration that this project has been able to pull off, and kind of, I'll be optimistic and say I hope 2024 is a good year, and it's looking like it's going to be. So with that, I guess I'll take questions.
Thank you, Mike. Any questions in the room?
Start you off with an online question.
Sure.
Okay, so we got a question from Ted Turner. They asked, it looks like nitrates were highest in Smith Creek, but productivity, as indicated by DO and pH flux, suggested not as high. Do you have any idea what's going on there? Is the water cooler due to spring influence, and is the temperature reducing productivity?
Well, I tell Ted, I think I said it in the conversation or the presentation. Haven't really gotten a good chance to look at this yet. So it's just the honest to God truth is there's a lot of interesting stuff on the first pass I'd like to talk about, but I haven't given it the time it deserves. So I know Ted. Tell him sorry, bud.
Sorry, Ted.
Yep.
Vanna, were there any other online?
There are not.
None? Okay.
There's a comment. Jennifer says, GMU researchers and students are very excited about being involved in this work.
Mike made a comment that we hope it's a good year, a good robust year for growth means probably not such a great year for.
I feel like that's brought up every year at this meeting.
Good year for who?
We need one, but yeah, it's great. Well, thank you.
Okay. Our next session is a lab session, group 2. We have 3 PIs who will be giving updates from their labs. Our first speaker is Margie Mulholland, who spoke earlier this morning, giving an update on the ODU lab for 2023. She's going to be giving you an update on sort of the year ahead. She had already kind of forecasted that, so she's going to bring up her slides in just a moment.
I think both Juliet and I thought that she was going first. Yeah. Darn it. Sorry, I thought the loaded. Recognize me that time though.
Prior years when we've done these lab sessions, it's been sort of an opportunity where the labs could share information and it's a little bit more relaxed. So this last hour of talks is really meant for the PIs to share information and for there to be a little bit more dialogue. So we have built in some extra time in between the talks for questions. So we want to make sure we take advantage of that. So 3 more talks in this last hour. So lots of time for discussion. Make sure everybody online and in the room bring your questions.
Okay, and I don't think I have too many slides because I really wanted to give a general overview of some things that we're doing in my lab that all pertain to HABs, really. We started out sort of trying to make a nitrogen budget for the Lafayette River once we realized it was a site for bloom initiation. And so I've had so many students over the years working on different aspects of these bloom projects. So here's our blooms that we're here for today. But so we've been looking at sources of nutrients to the estuary and even looking at coastal flooding as being a source of nutrients to the estuary, looking at runoff, rainfall, trying to understand the biological controls on bloom formation. And Eileen Hoffman, one of my collaborators, has made a lot of progress on making a biological model for bloom formation. And then we're also looking at a lot of different physiological factors that affect bloom formation as well as success. And so you'll see little red names underneath these things. And these are different people that have been associated with different aspects of this project. And I'm not going to show you data associated with everything because it would take hours. But so I thought putting in this flow chart was a good way to do it. But we've had a lot of undergraduate REU students involved. Mike Echeverria, who's a senior graduate student, is trying to finish up, and he's looking at interannual variability of blooms. Katie Kreider, who you heard from earlier, she's actually working a lot on mixotrophic capabilities of algae and molecular controls on that, seeing if we can find genes that are related to mixotrophy. And Eduardo Perez Vega, he's been with me quite a long time as well, has been looking at cyst dynamics because these algae have a lot of behaviors. They have complicated life cycles, and they have resting stages that reside in the sediments. And so we think sediment resuspension plays a role. And so we've worked a lot with Rip Hale on sediment suspension and deployed some equipment. Actually, some of the data I'm going to actually show has to do with some of the work we've done with him, because in previous years, we found that the sediments not only are a source of cysts, but whenever we get resuspension in this really shallow microtidal estuary that initiated, is we get a huge bolus of nutrients coming up. Because the sediments are just a reservoir for all this organic matter that's just sitting there degrading. So we've been working a lot with Rip Hale on that. And we haven't looked so much at the top-down controls, just a little bit, but we are looking a lot at climate variability and change. And so actually last week I was in New Orleans giving a presentation on heat waves in estuarine environments and how those affect blooms. And we think they can actually, depending on their timing, they can both be a positive and a negative effect on blooms. And so anyway, I just wanted to go through a lot of the sort of different projects that we're working on and still working on. And then a new faculty member, Joe Temborski, we're also working on groundwater inputs. And I know he's working up here in the York River as well, because that's a major source of nitrogen fueling blooms in these coastal waters. But anyway, that's just sort of a broad overview of what we've been doing in the lab. I probably missed some things there, but we're also working with the NOAA team on modeling. And as you saw earlier from Katie, we're working with Victoria Hill, who was here earlier today, but they had to leave to go teach. And so, but we're working with her on the planet.com data, trying to look at high resolution images of blooms and to try to even get a carbon number. They've got an algorithm now that they can calculate, they can convert that chlorophyll to carbon and try to figure out how much carbon are these blooms responsible for, because that's what modelers are very interested in. And so we're doing a lot with the bloom data. And again, like I said, trying to relate the blooms to meteorological forcing, because again, this surveillance is important. We can get out on the water here, but like this past summer, we had a bloom out in the main stem of the bay, and it was really hard to get responsive sampling out there. And so we want to get better and better products so that we can really know what it is it harmful? Do we need to worry? And I did say I'd show a little bit of data, and this is just some of that sediment resuspension data. It was done by an REU student. And after the wind blew steadily for a little bit, we went out and did vertical profiles, and there are huge concentrations of ammonium that go into the water. And this is 60 micromolar. So that's a really big number for the estuary. And similarly, we saw that cysts got resuspended after the sediments were resuspended. And we think that that's really important for bloom initiation. So we're working. How did you initiate the cysts? Microscopically, we looked at it. Yeah. And they weren't particularly Margolophodinium cysts. Yes. Yeah. They were just cysts. So, yeah. And so for this experiment, we just did stirred and unstirred sediments. And of course, when you stirred them, the cysts came out. And actually dinoflagellates came out subsequently as well. But it turned out the RU student working on that wasn't very good at identifying things under the microscope. And so we generified the project a little bit. But that's, again, one of the challenges we have with undergraduates and summer students that aren't around very long. But let's see. Yeah, and then this is the more fancier sediment resuspension study we've been doing, and we have a manuscript that's in revision on this one. And this was in collaboration with Rip Hale. And here he deployed some fancy instrument, an aqueduct that measured current velocity, and we could see these periods of wind events followed by spikes in nutrient concentrations, followed by our bloom initiation. And we were able to repeat this. Not always, not every event did that, but a lot of events did do that. And so we've have a link for bloom initiation. And we care about the initiation because we understand it. Maybe we could stop blooms from forming in the first place, especially things like Margolepidinium, that there seems to be hot spots for where they initiate. Okay. Oh, another project has been looking at water quality impacts from coastal flooding. And I have a recent graduate student that Alfonso Macias Tapia did a lot of this work. He's now working in the NOAA Education Office. He was a Knaus Fellow and then was hired on. So there is hope after graduation for jobs. But we have a lot of blue sky flooding down in Norfolk. And those of you from Richmond may not see a lot of this, but almost every spring tide now, this is what our roadways look like. And I know that they weren't counting this in the Bay program models that are establishing the nutrient loads for being protective of things like HABs. And so we were able to leverage a citizen science project going on and collect 200 samples within an hour of high tide. And we were able to calculate that the nutrient loading from coastal flooding during a single event, the entire annual load of nitrogen was delivered to the Lafayette River, which it has its own TMDL. So we were able to reproduce this 5 years running. And it's something that we've not been counting on, this particular climate change impact that's related to HABs and HAB formation. So in addition to the heat waves, we have that. And then finally, I think this is my last slide. I just want to tell you all a little bit about our HAB project and our goal to increase surveillance and to transition this whole program to management, where it's just inherent in our observational program and early detection system. But essentially, we just had funded a regional HAB grant. And the idea behind that is you all saw a lot of the fancy flow cam imaging data earlier and IFCB data. Well, IFCBs and flow cams cost about 130, 150K. Okay, is Virginia going to buy 9 or 10 of those to put in our waters? No, not a chance. So the Planktoscope, which Savannah mentioned, is a low-cost imaging system. They cost about $3,000, a lot more manageable, right? So as part of this program, we're going to enhance our observations by building 30 of these Planktoscopes, and we're having a demo after this this session today to show you what the Planktoscopes look like. We've ordered our first 12. They're supposed to get here this week. And so we'll be hosting a building workshop. And what we want to do is deploy these with end users to increase surveillance. Over the project period, we plan to build 30 of these and deploy them with partners around the lower Chesapeake Bay to surveil initially for Margolepidinium and Alexandrium. That's in the proposal. But really, we're training our classifiers to analyze and identify all of the HAB species in Virginia. So we'd love people to run these things year round, and they're really easy to use. And then we can patch the data through to a platform called EcoTaxa, which we're using to annotate the databases. And that's handy because we could potentially develop an early detection system in near real time and get the word out to users so that people can avoid the water. So we're really excited about this. And as part of this, we're also using observations and some experimentation to advise models. And we have a biological model that we're embedding in a hydrodynamic model. And Dante, who spoke earlier, is involved in that. And Eileen Hoffman is involved in the 1D modeling. And the idea is to develop a regional HAB forecast that we can get the word out, like I said, to the different users group. And we'll continue to use our existing surveillance tools but just try to expand those. And we're deploying a QR code for users that hopefully can then also, in addition to reporting to VDH, report to us when they see discolored water, so we can get a water sample and feed it to the planktoscope. You know, we need observations. So I think I addressed most of that. But again, if you can stay and see how the planktoscope is used, and we'll be having a later workshop, and we're building the 30 and giving them to our users. Okay, so you don't have to buy them. And this is our image. This is the platform we've been using, and a lot of the work for me right now is trying to reconcile the different imaging platforms. We have IFCB, and there's a dashboard that a lot of that data goes to and it resides in, but it can accept data from the flow cams and the planktoscopes. So the nice thing about EcoTaxa, it's open software. It's right now it's maintained by the French government, but it ingests data from IFCB, flow cams, and planktoscopes. Okay, so it ingests all of them, and then it's very highly functional, so we can use their AI to train the databases to get better and better at identifying the target species. So we're really excited about that because we're involving our regional IUS system. For those of you who don't know what that is, that's the Ocean Observing Systems, and we have a Mid-Atlantic regional one, so that's called Maricus. And VCUS, the Virginia Estuarine Coastal Observing System, is hosted here at VIMS. Willie Ray does a lot of the work on that. But anyway, it's been a real headache trying to figure out these multiple systems because these imaging technologies are sort of coming online really rapidly now, and everyone has their own and wants to use their own. And so they're not networking really well. And the IFCB dashboards, the California Observing Systems has 9 IFCBs deployed, and they're all the data is going to the dashboard for those systems. But like I said, I don't see Virginia investing a couple $1 billion in deploying IFCBs that need to be maintained and everything else. So these low cost observing systems, I think, are going to be the way to go in the future. And I think they'll become lower and lower in cost. The first one we built, it was just with parts that we could buy on Amazon. And if you go to the planktoscope site, it'll give you like the part numbers from Amazon. And yeah, you've seen pictures of the libraries and whatnot. So, and I think I said all this. Oh, but another useful thing that's in these classifiers that you can see, and I think Savannah and Kayla may have alluded to it, but we can see different life stages in there as well. So we can do work on cysts. One of my students have been looking for cysts with the flow cam images. So you can do a lot of different things. And I think I said all that already as part of the last one. And I always like to have my New Year's resolutions. It's a little late, but our plans for the next year, things we plan to do is, well, work on those long overdue manuscripts. We all have many of those. And so we'll keep doing that. We've been able to do more year-round sampling and analysis, and we're incorporating lots more undergraduates, especially this year when I'll be starting to teach a CURE class, which is a classroom-based undergraduate research experience. And we can put students on this. We actually have IFCB planktoscope and the flow cam, as well as the microscopy. So we're planning to sort of sync all the different types of measurements to make sure we're always measuring the same thing, whether we're using one imaging system, another imaging system, or looking at it under the microscope. So that's in the works. We're going to deploy the IFCB at our time series station in the Lafayette River. And we'll also tap into, there's an IFCB going in as part of the Pioneer Array. I don't know how many of you are familiar with the different ocean observing platforms that exist, but the Pioneer Array was meant to be a mobile array that was developed by the National Science Foundation, and it is moving to right off our coast to the area of the Hatteras Front. And so it'll be, I think, a really useful tool because we've seen Karenia blooms emerging in some of the samples that we've been collecting in recent years. And I don't know how many of you are familiar with Karenia and Karenia brevis, which causes a nasty HABs in the Gulf of Mexico. But we've seen Karenia cells and blooms in the bay now. And so we're very concerned and seeing them in the Mid-Atlantic region. And so that IFCB that's on the coast, on the shelf, that might be a good early detection system for potentially emerging species that are getting into the bay mouth now as a result of climate change and changes in circulation. And let's see. We'll continue on with the Lake Anna study, and we're going to try to isolate cultures to do some other experiments with, where Aaliyah is really focusing on looking on the role of nitrogen fixation in these blooms, because we think if we've shifted the nitrogen to phosphorus balance in the freshwater systems, that some of the cyanobacteria blooms that are emerging could in fact be nitrogen fixers, and many of them are, we know, we see their heterocysts. So, and Katie is working on the mixotrophy aspect, like I said, and looking at life cycle events that influence blooms. And I think I had 2 pages of resolutions. I had a lot of resolutions this year. And again, we want to build and deploy all those microscopes, develop that forecasting system, and align those platforms. That's all a part of the Mayor HAB. I doubt we can get through all of that this year, but we do plan to build and deploy 12 of the microscopes. So hopefully spread the word. We want people to use them. We'll continue our modeling experiments with Eileen for the biological models, and then getting her model embedded in Margie Friedrich's hydrodynamic model. We have those several aligned projects that I talked about at the beginning that we'll continue to work on. Is that nitrogen budget for the Lafayette River, trying to figure out how long the nitrogen stays there once it gets in there and sinks to the bottom. We'll look at sediment resuspension and its role in delivering nutrients as well as cysts into the water column. We'll continue to work with Stephanie and Joe on the groundwater inputs, and work on that planet data with Victoria Hills Group. And we're continuing on with our coastal flooding and trying to relate that with subsequent blooms as well. As Ben, a new graduate student, is looking at the planet data and correlating that with when there's been a big flood delivery of nutrients. And Alyssa is working on GIS systems, that she's interested in how land use affects the coastal flooding and the nutrient delivery during flooding. So got a lot going on. And I, of course, am not working in a vacuum, so I don't really do anything. I just talk, and everybody else does the work. Okay, any questions? And I didn't get the light flicked on me.
Thank you for the exciting talk, Margie. I have a quick question. I think it's quick. So the coastal flooding stuff, I was really interested in that. And you said 200 samples were collected. And I was wondering if you could, I have no idea how that kind of study works. So I was just like, how do you do that? And
well,
200 samples.
Well, it's actually a funny story, because when this all started with, they developed a phone app called Sea Level Rise. And actually, when there's coastal flooding, you tap the phone, tap, 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 and make a map around where the water line is. And the data, gets ingested into Derek Loftus, who's here at VIMS, some of his models to verify them. And in 2017, I knew about the phone app way early because it was a nonprofit of my spouse's that developed it. And so we would go out and beta test it. And that's when I knew that there was a lot of literally shit going in the water when it flooded. And so we got to thinking in 2017, they had this huge community effort to build awareness of coastal flooding. It won a Guinness World Book of Records for the most data points collected during a citizen science project. But the public media station, Wavy TV 13, and the Daily Press all advertised for this big Catch the King event. And that was a king tide event, is what happens during a perigean spring tide, or when the moon is closest to the earth. So they usually happen in the fall. And they advertised and advertised. So they had thousands of volunteers come out, or hundreds come out to map. And so we knew they were going to map on that day. So my kids were in middle school and high school, and I knew they needed volunteer hours for school. And so we got all these volunteers from the Norfolk Public Schools, as well as graduate students, and we trained them to go out and we put them in groups of 3 or 4. We trained them to map and to collect water samples for us. And then we processed them. We had them bring them back in coolers. We gave them kits and brought them back to ODU. We actually have a proposal in to try to expand to the York River and probably the Choptank in Maryland. Yeah. But it's a long story.
I'm Margie. Have you done any like direct comparisons between the planktoscope and the flow cam? I mean, they're both taking pictures of things, and I assume that there's something the planktoscope that the flow cam does that is better than what the planktoscope does. Yeah. So could you elaborate?
I think they collect more metadata and, more optical measurement size and lots of, Savannah would know what all the different parameters. So they measure a lot more things about the cell. The planktoscope just images, but if you import it into Ecotaxa, you can start to do some of those things. We had a paper come out in 2023. Sophie Clayton was the lead author, and we compare the different imaging tools. But the plan is to compare all the different imaging tools with each other, as well as with microscopy. And so right now we've been in the process of counting cells. Then we give them to Kayla to run on the flow cam. And then as soon as we build the first new planktoscope, we'll run them on that. And when we get the IFCB running, we'll run them on that. So we have both preserved as well as live cells. We can do comparisons because we're finding we need to build both live and preserved libraries for the images as well. Is that, you might have more details for the.
Do you mind if I?
Yeah.
Okay, so
The planktoscope, I think, shouldn't be a monitoring tool in that it's not used for quantifying very well.
But it can be.
Can be.
It can be. I've talked to Thibaut and Adam.
Okay.
Yes.
Maybe the newer software will help make it better for quantifying, but I guess right now, or older versions of it, it's good for taking a look at what the plankton community is like as a whole, but there's some fine-tuning, a lot of fine-tuning in the software and hardware that needs to happen before it's ready to be quantifiable.
Right, because how it works is you go through the flow cell, it takes pictures, but they're not all in focus sometimes.
Yeah.
But it works much like the images of the flow cam that they showed. But you can calculate the volume that you're imaging at any one time so that it can be quantitative. And and they have used it quantitatively. Yeah.
Margie's demo is right after our task force meeting, just down the hall. So you do need to be in person for this. So those of you that registered online and are not here, you can reach out to Margie and she will. Maybe you could talk a little bit about, do you have any other demo workshops planned?
Well, the next workshop we plan is the kits are supposed to be coming this week. And so we plan to build our first one next week of the newer version, just so we all know. And then what we want to do is identify users that will then we'll have another workshop that will build them together. Because it takes a couple hours, they said, to build one. And the idea would be then to have the users the whole process be demystified for the users so that in the field, the user isn't afraid to swap out a flow cell or, swap out a camera lens, because they'll have seen it, they'll have built it. And Thibaut and Adam are the people that invented the planktoscope. And they said you could probably build a new one in 2 hours or less. But again, if we could do it end to end and learn how to upload the data then into the Ecotaxa, because that's another little piece. But we give the users tablets too, and they come in little Pelican cases. So, you can take them in the field or, you could use them in the lab, either way. But you need internet to pipeline the data to Ecotaxa.
So your planktoscope to build them. I guess those materials are arriving this week. Do you have another workshop scheduled or on the books?
It's not on the books, but ideally we'd like to have it in April sometime. Just so they're built and ready to go before.
So people should just email you if they're
Yes.
interested.
Yep.
And
You
you'll
can
put
send-
them on the
Yep.
a list.
I'll put them on the list.
And anybody that's here and that hasn't registered, there's definitely still space if you want to stick around.
Yeah, we have a group meeting for the Mayor HAB project on Friday. And so that was one of our agenda items, is to set that date.
And the workshop will be in AB.
Classroom AB, right down the hallway.
Yeah,
Any
and
other
it probably will work for freshwater systems too. So, there's no reason not to want to be a user and be a freshwater person. Even though this project is aimed at providing them to people that would be in the lower bay and coastal bays. And yeah.
Any other questions for Margie?
Given their $3,000, you could buy one for Christmas.
In whose world?
Yeah. Okay. And if you buy the parts on Amazon, I think you can get them probably for under $2,000.
Oh,
Yeah, because it's even less now, because some of it is to buy the tools, and once you have the tools, you only need to buy that once. So, but it's just a pain because they're not all in one place, and you have to go get the casing fabricated. And, yeah.
So
Yeah.
Oh, Margie, can you hand her your microphone?
I'm not holding. I'm just talking.
We're-
about
okay.
getting from done.
Oh, you're so loud.
Sitting right below me. So if I moved my head, you couldn't hear it.
There's a quick comment for the audience. It says, please tell Chris Jones from GMU to buy us for Christmas.
We buy this one too.
Ben, I don't think he heard you. There is a comment for Chris Jones.
To buy one.
To buy one for GMU. Did you get that, Dr. Jones? You've been requested to buy a plankton.
All the parts. I just need the,
I didn't get the casing built, so I'm gonna come join you somehow.
Yeah.
Okay.
Sounds good. Okay. No.
Okay, Dr. Smith is associate professor here at Ecosystem Health at VIMS. She investigates the chemistry, ecology, ecotoxicology of bioactive compounds synthesized by
We have a picture of Sarah in the corner.
HABs in freshwater, estuarine, and marine environments. So she's going to talk about what they have upcoming in 2024.
Thanks everyone for giving me a chance to talk about some of the stuff our lab's been doing. I'm going to start off by saying that this presentation is going to focus a lot on dinophysis, and that's because we currently have an EcoHab that's in its 5th year. And so we're starting to see enough now that some of the work is starting to weave together into a story, and that's what I'm going to start to walk you through today, which is really exciting for us. But to be honest, our lab does not discriminate against any toxins. We enjoy studying all of them. And so we have projects that span many different lipophilic and hydrophilic toxins. But this one's going to focus largely on dinophysis. So what I'm going to do is tell you about a few studies that are either just published, in preparation and currently with co-authors, or are currently in revision. And kind of weave those together for you. The first one is going to focus on really trying to understand the effects of dinophysis and its toxins on oyster larvae. And another one is going to talk about, instead of effects of the HAB on organisms, the effect of the environment, which is in this case turbulence, on the HAB. Okay, so backing up, we work again with multiple toxins. So if you don't see your favorite on there, please let us know and we'd be happy to add it. But that's going to be your dinophysis toxins, your oxalic acids, pectinotoxins, yezotoxins, azasparacids, domoic acid, or your AZTs, ASTs, your microcystins, brevetoxins, saxitoxins. Those are some of the current toxins we're using in the lab. And we also have another study that Marta is leading up, working with bioactive chemicals. So not something that is quite called a toxin.
at this stage, but does have bioactive properties. And for us, that right now are the oxylipins and the PUAs, which all you need to know there is those are oxidized products of PUFAs, or polyunsaturated fatty acids. So we're looking at the effects of all these different chemicals on a few different target species. Currently, right now, the work being done by Marta and Noor is focusing on oyster larvae. But Josh, who you heard from earlier, is bringing us into shrimp, specifically white shrimp. And Isabel is bringing us into blue crab, and Olivia is bringing us into fish. And we do this both at like a bioassay level, where we're in, small micro mills, and also in a jar level. And then Olivia might be taking us up to the level where we're doing it in mesocosms in the seawater research lab. Oh, let me move this. Here. All right, so jumping into that first topic, again, this was looking at thinking about the effects of HABs and their toxins with the specificity on dinophysis and their effects on larval mortality. So I'll start off by telling you that Dinophysis acuminata from Chesapeake Bay produces two classes of toxins. The first one we've been hearing about during the day today, which is okadaic acid and its Dinophysis toxins. You've been hearing about those because they are regulated in seafood in the US and the EU and many other countries. But we haven't been hearing about our pectenotoxins. So pectenotoxins are another group of lipophilic toxins that are produced by Dinophysis, but are not regulated in the US. And they have recently been deregulated in the EU. And that's for good reason. We shouldn't necessarily be thinking about them for the effect of human health. But they do seem to be important for animal health and shellfish health. So I really want you to be thinking about those two groups at this time. And I can tell you that this is a Dinophysis isolate from Chesapeake Bay. It's from Nassawadox. And this Dinophysis in culture produces this toxin profile. So this is pectenotoxin 2, which is just one of the pectenotoxins. And then this okadaic acid and DTX1, these fall into that diuretic shellfish poisoning toxins. So from the perspective of an organism being exposed, they're not just getting the ones that we care about, they're also getting these pectenotoxins. Backing up, I can also tell you that Dinophysis acuminata is typically found in Chesapeake Bay. I think Walney found it in 2020 to be an average of 0.4 cells per mil. But we had talked about how Dinophysis is a low bloom species. And so a bloom of Dinophysis acuminata is considered anywhere between 1 to 1,000. And to be honest, just 10 cells per mil in some states does trigger shellfish monitoring and even closings of harvesting beds. So Dinophysis, you should be thinking of levels of 10 cells per mil, not the really impressive hundreds of thousands of cells per mil that you've been seeing in some of the other blooming species. All right, so then came along Sarah Pease, who's hopefully still online. And Sarah, as a graduate student, really wanted to start to understand the impacts of these cells and toxins on the eastern oyster larvae. And so with an eye towards hatchery and maybe survival of the fishery, she really wanted to focus on the younger life stages. So she focused on 7-day-old larvae. And in this example, she did a 96-hour exposure. So I'll try not to get too far into the weeds here, but it's a really cool study where essentially she exposed the larvae to different cell concentrations of Dinophysis acuminata. And she did that over taking time points from every 24 hours for the 96 hours. And she measured both inactive and dead. So how many larvae became inactive, they're not swimming, versus how many were actually dead. And so what you can see here is that when she gave them the highest amount on that range of what's considered a Dinophysis bloom across the world, when she gave them the high level of 1,000 cells per mil, she did find significant mortality. And you can see the trend that they're inactive, they're not swimming, they're not doing well, and the next day, a portion of them are dead, right? And we see that pattern over time. So this started to indicate to us that there's something just about the cells that are causing this effect within the larvae. So then Sarah moved to pure toxins. And so here you can see that she took a high amount of toxin, no doubt, but it was just to see if this could actually cause an effect. And she took a high level of okadaic acid, again, the ones that we regulate for in seafood for protection of human health. And she exposed just the pure toxins, so no algal cells,
again to these 7-day-old larvae. And what you can see is nothing really happened with okadaic acid. And so this is equivalent to 10,000 cells per mil. So even though she went an order of magnitude more than what we considered even a typical bloom, this would be a peak bloom that happens in Argentina, right? And so even by doing this high level exposure, no measurable effects on larvae. However, with PTX2, she had to end the experiment within 48 hours because there was so much death. So this really started to point out to us that pectinotoxin 2 might be responsible for shellfish health. And it has kind of put us on a path ever since. I will point out, though, before we leave this slide, that Sarah also tested the 10 cells per mil. So that's kind of the number you've been hearing thrown out about the maximum number of cells we're really seeing of dinophysis per mil within the Chesapeake Bay and its coastal bays. So she went back and tested the 10 cells per mil as well, and she was not seeing a measurable effect on the larvae survival, which is important to know. So this suggests that at our bloom levels, there is not a direct impact or measurable impact yet of dinophysis on larval survival. However, during some not random, but high peak concentrations, you might find that effect. All right, so next comes in Noor. And Noor decides she would like to build on this study, but now she really wants to go into that 10 cells per mil, right? So I'm going to slip you back. So 10 cells per mil, this realistic level of a bloom in Chesapeake Bay. She then continued the 10 cells per mil forward, but she did 4 different species of dinophysis. There are, I think, a dozen toxigenic species of dinophysis, so she took 4. And in collaboration with many, we had 4 isolates of dinophysis. So right here, you have Dinophysis acuminata here on the bottom. That's the same species that Sarah had trialed. Noor brought in Dinophysis ovum, Dinophysis 40i, and Dinophysis caudata. At 10 cells per mil, she then exposed them again to the 7-day-old larvae for 48 hours and looked for a response. I can point out she did a couple different treatments that I'm going to lead you through. She did live, so that's equivalent to just live cells. So the cells are swimming around with the larvae, they're interacting, and she is looking for mortality. She also did lysate, which is taking the cells and exploding them. So you're getting, the larvae are getting exposed to anything that was within that cell, right? So that's lysate. And then Noor applied this additional treatment, which was dialysis membrane. And so for that, Noor had taken a membrane that was 10,000 Daltons. And the dinophysis goes in the membrane and the larvae go on the outside. So this is a way to separate the larvae who are swimming around happily on the outside to the dinophysis producer of toxins in the inside. In theory, she picked a membrane that is so large in pore size that the toxin should be able to move back and forth between the membrane and the outside water. So it allows the larvae to still be exposed to the chemicals of the dinophysis without actual physical contact with the dinophysis. It is important to point out in all of these studies, which I haven't said yet, that dinophysis cannot be eaten by the size of larvae. Sorry, I forgot to tell you that. So any response that we're seeing between the larvae and the toxins or larvae and the dinophysis must be due to the chemicals. These larvae are too small to eat this large of a cell. All right, so taking you through some of these results, the biggest take-home thing from Noor's, biggest take-home point from Noor's work is that contact with live cells was needed for toxicity. And toxicity did occur at 10 cells per mil, but not at all species. So again, Dinophysis acuminata at 10 cells per mil, just like Sarah had seen, did not lead to any noticeable mortality compared to the control, where they were not exposed to any toxins or dinophysis. But with 40i and D. caudata at 10 cells per mil, the live cell treatment did result in significant mortality in the larvae. So this starts to suggest that the cells, being that they're too large for consumption, there's chemicals that are causing this effect that are specific to 40i and caudata at 10 cells per mil. Now that could be the toxins, or it could be bioactive endogenous compounds or BECs. So other chemicals that are allelopathic that we have not yet identified. what you need to know now is that Dinophysis fortii and caudata produce significantly more PTXs. So again, this is leading us to the point that Dinophysis ovum, for example, that only produces okadaic acid within the cell, was not toxic to the larvae. Dinophysis acuminata can be toxic at 1,000 cells per mil, according to Sarah's paper, but at 10 cells per mil was not toxic because the proportion of PTXs was not high enough, or the amount of pectinotoxins was not high enough. However, once you move to fortii and caudata, those cells are producing a lot more toxin per cell within each cell. So that leads us down to conclude that the cells with more pectinotoxins are more lethal to those larvae. Noor also went ahead and tested again the 10 cells per mil level of toxins. So she tested orders of magnitude pure toxin less than Sarah did. So Sarah was just a proof of concept. Noor came in and was like, I'm going to dial this in and take it down to 10 cells per mil equivalent. So she exposed the larvae to the toxin at lower concentrations and still found again that at the amount of pectinotoxin equivalent to 10 cells per mil of D40I, that pure toxin led to the effects. So it does suggest that yes, there may be other bioactive endogenous compounds within D40I and within caudata that are causing the larval death, but pectinotoxins is also playing a pretty big part. So I will point out that this does start to link to some of the stuff that Marta Sanderson led, where with collaborations with Kim Reese and a bunch of hatcheries, we have found that okadaic acid and pectinotoxins can break through into hatchery waters used for rearing larvae. So these are waters that have been treated and we're still able to find, so they've gone through filter, UV, and different types of carbon filtration, and we're still able to find toxins present whenever we look of okadaic acid, DTX1, and pectinotoxins. So there are some possible impacts that could be assumed that are happening to survival or increased stress during a bloom event. I'll lead you really quickly through this one. There's no figures, but I wanted to let you know that that Vanessa had also worked with Dinophysis, and she took 4 strains of Dinophysis and exposed them to turbulence for 6 days. We tried to recreate turbulence using an orbital shaker. These are the 4 species, acuminata, ovum, and caudata. And she had 3 treatments: no turbulence, so think of this as like still control, turbulence, and high turbulence. And what she was really trying to look at is representing inlet, estuary, surf zone levels of turbulence. And would that cause Dinophysis to not be able to feed on its prey, or would it impact its ability to grow or produce toxins? Just to give you an idea, this is where all of her experiments fell in regards to the amount of turbulence generated. So you get to see that she's falling in within this realm of surf zone, inlet, continental shelf, and at the upper end of open ocean. She measured ingestion rate. So Dinophysis must feed on prey to grow and produce toxins. So she wanted to see if turbulence would essentially interrupt that feeding mechanism. She looked at growth rate of Dinophysis and then the intracellular and the extracellular toxins. And this is the summary table because it's easiest for me to explain through this, but this was her graphical abstract. So as turbulence increased across her 4 different species, she found that in general, the 2 acuminata strains and the Dinophysis ovum decreased their growth rate. So what that means, in general, as a system becomes more turbulent, maybe after a mixing event, or you're getting near the shorelines and you have more disruption, Dinophysis acuminata and ovum are going to slow their growth rate. Interestingly, they increase the amount of toxin they have in their cell. And this decoupling of growth and toxin content has been found for many species. This is essentially the idea that as cells keep growing and dividing and dividing and dividing, they kind of keep a constant level of toxin. But then all of a sudden they reach kind of this peak of growth. In this case, their growth is being slowed by a stressor, and the cells get big and fat, and they hold on to that toxin because they're not dividing and making daughter cells.
So it does suggest that in certain areas where turbulence could increase, you'd have a decrease in growth rate. The cells are going to become more toxic. I'll end on the interesting idea that Dinophysis caudata, however, seemed to be stimulated by turbulence and its toxin content went down. I'll end there. Thank you so much for listening. Hopefully you saw some interesting things about Dinophysis, and these are all of our great collaborators, and my email's at the top. Thanks.
Thank you, Juliet. Our last speaker of the day is going to go ahead and bring up her talk. Kim Reese is going to round out our talks. While she is bringing up her presentation, do we have any questions for Dr. Smith?
I'm going to keep my phone.
Any questions in the room? How about online? I'm guessing not. Nope. Again, our esteemed host is going to round out our talk. She's going to talk a little bit about what the Reese Lab is going to be up to this week at BIMS, or I'm sorry, this year in 2024.
Well, actually, I think I'm going to give a summary of what we've been doing and kind of what's going to be coming out from things that have been done. Can you all hear me? Okay, good. Wake up. Yeah, I know. It's just after lunch. So I'm going to go through sorry for those of you who've already seen this many times, but this is kind of Margie, this is my resolution for 2024. We've got to get this published, but I want to put this as a background because this kind of sets the stage. This is work from Clara Robison, my former grad student, her work. But this is just to say, what are the impacts? Remember those large blooms of Marge and Alex that happened in the late summer? What happens to oysters that are deployed out in the field when they are exposed to these blooms? So Clara did several years field study, and it turned out that the year we had very large blooms of Marge and Alex was also the year we had a pandemic, okay? But we had a very large bloom that year, and we did happen to try to get out, and we did the best we could at getting these data. But what we saw when Clara ran all the models, we saw that the growth rate is lower when Marge or Alex concentrations are higher, and that that impact lags the cell count by about 2 weeks, which is consistent. So you can see here, this is the shell height and the change in shell height over time. And you can see clearly as the Marge and Alex blooms start, the change in shell height decreases dramatically here as those blooms occur. Okay, and that's just to show you what's going on. The other thing that happened, and this is important, and this was building on work that Sarah Pease, that Juliet just talked about. She had done work in the lab when she was a grad student doing her master's work. She had exposed adult oysters in the lab to cultures of Alexandrium monoletum, and she had seen the toxin made by Alexandrium monoletum being taken up by those oysters while they were being exposed to the culture of Alex. And so that toxin, the primary toxin, is Goniadomin A. And so she saw that toxin being taken up into the oyster tissues. So what we did was look at what happened to the oysters that were out in the field. Did they, while they were being exposed to the Alex bloom out in the field, they take up the toxin as well? And this is done in collaboration with Juliet's lab. Juliet and Marta were involved, and they ran these tissue samples after they were extracted. And what we see is indeed the Goniadomin A, the GDA, it'll go by that initial in this talk. GDA is accumulated in those oyster tissues when those oysters are exposed. And then after the bloom dissipates, so these are July and August samples. These are the oysters, and these are the oysters during the bloom here in 2020. And we see that the oysters have taken up the GDA, and then by October, they depurated that toxin, okay? So then, coming into our lab was Sylvain Gaillard, and he did a great series of experiments to kind of follow the fate of that toxin, that GDA, that's produced by Alexandrium when it's taken up into the oyster, and what happens to it in the oyster tissues. Because we know that there are different forms of this toxin. And we know this because of a lot of the work that Tom Harris, who's sitting here in the audience, he's an organic chemist from Vanderbilt who came and worked at BIMS for several years. And he was the one who isolated GDA for us and did a lot of the characterization of these different congeners. And we know that there are these different forms. And so Sylvain and Hamish did a lot of the work to follow the various congeners in the oyster tissues. As the oysters took up the GDA, we were looking to see, sorry, got to move this around so people can hear me. We followed to see what happened to the GDA as it was taken up, as the oysters were filtering Alex, the Alex culture in the lab. The GDA was taken up into the oyster, and then we followed to see whether it was being converted in the tissues as it was transported through the oysters tissues. So Sylvain exposed the oysters to a sublethal amount of Alex for 96 hours, and then followed them for another 96 hours for depuration. And he had controls that were unfed oysters and controls that were fed on non-toxic phytoplankton, basically oyster food. So 2 sets of controls, and then ones that were fed the 615 cells per mil. And so what happened? So the oysters that were fed Alex, indeed, GDA was taken up very, very early and was observed in the gills of the oysters, which is what you would expect, because as the oysters are feeding, they're taking up these Alex cells over their gills and absorbing that toxin to their gills. So right away, you see this GDA, the Goniodomin A form, right away at 6 hours in the gills. You see a little bit of the other forms, the GDB and GDC, and some of the secoacid form as well. And you see that continuing at 24 and 96 hours in the gill. What was interesting is that at 24 and 96 hours in the digestive glands, we started to see a conversion of the GDA over into this secoacid form. So it looks like in the digestive gland that GDA was being converted over into that secoacid form of the toxin. In remaining tissues, it seemed that primarily the GDA remained in the GDA form. And we saw just a little bit of the other two, the GDB and the GDA forms of the goniodomins. And during depuration, there was relatively rapid depuration of the toxins during the depuration period. The other thing that Sylvain did was he looked at the impact on the gene expressions in the oysters for some targeted genes. And the genes that we were looking at were structural genes, in particular actin, because from previous studies that had been done a long time ago for looking at the impact of goniodomin, they had looked to see what would happen if you exposed animal cells to goniodomin, and they had seen an impact on actin. So we were particularly interested on looking at the effect on actin gene expression. And indeed, what we saw was an effect on the gill. You see the actin genes are upregulated when the oysters were exposed to goniodomin, to GDA. The other thing that we observed was some of these other stress genes were upregulated upon exposure to GDA, to Alexandrium. And there's several others here as well. And that continued for we had several time points here that I'm not going to have time to go over, but there are, and even into depuration, there was still upregulation of some of these stress response genes and actin. The other thing, I don't have a lot of time here, but the other study that Sylvan did, and we're just now getting this manuscript in prep, was looking at allelochemical interactions of alexandrium with other dinoflagellates and diatoms. We were particularly interested in this because of the fact that we see often marge bloom before Alex. And so one of the questions has always been, we've often seen this, that when you put marge with Alex, that marge goes to the bottom of a flask when you mix the two organisms together in the lab. And one of my techs used to say that marge will go down there and pout. Basically, it stops swimming. It becomes immobile. So Sylvan did a series of studies looking at the effect of the Alex on marge, Margolophididium polycricoides, Chaetoceros, and Akashiro. He looked at mortality, motility, morphology, growth, and photosynthesis. And I don't have nearly enough time to go into the details on this study because there's a lot that was done during this. And he looked at culture supernatants from 2007 and 2020. So those are two different cultures, one that we established way back in 2007 and another that he established in 2020. And then the purified toxin, Goniadominae, and purified Goniadominae secoacid. He did all of those exposures. And what basically the bottom line was, the 2020 culture is more toxic than the 2007 culture based on these exposures to Chaetoceros. It caused inhibition of photosystem 2. So we saw inhibition of photosystem 2 in Chaetoceros. We saw mortality of Chaetoceros that was clearly higher. And as the increasing concentrations of Alex, we saw increased mortality of Chaetoceros. And we saw altered cell morphology through flow cytometry based on cell size and cell complexity, which was measured by side scatter, if anybody's interested. And the secoacid concentration was twice as high in the 2020 strain, even though the GDA concentrations were similar, if anybody's interested. Interestingly, for the supernatants, Akashiro, we saw no effect on motility, photosynthesis, growth, or cell morphology. And for marge, we saw no significant mortality, but we did see inhibition of photosynthesis. Motility was inhibited. There was no effect on growth. Cell morphology was in fact impacted, however, by the Alex 20 culture, not by the 2007 culture. Oh, okay. And for Chaetoceros with GDA and secoacid, we saw no impact on mortality, growth, or photosynthesis. Cell morphology was affected by GDA, but not by the GDA secoacid. But that's the only thing that was affected, was the cell morphology for Chaetoceros. This is evidence that there are other allelochemicals in the supernatants of Alex that are responsible for some of the effects that we're seeing. And the same thing with marge. GDA inhibited the photosynthesis and impacted motility and affected morphology. And only a little bit, you only see the highest concentration has an impact. And this is much lower than the concentration that we saw. And only the highest concentration of secoacid affected morphology. So we have clear evidence that there are other allele chemicals in the supernate that are involved in what's going on and how large, how Alex is affecting other organisms. So there are other things involved other than Goniodoma toxins. So this is a summary, but you can read that because I don't have time.
We have time for a few questions for Dr. Reese. Any in the room?
What's the impact on morphology, like for them? What's the impact on morphology for the Margoliphididium? Are they shell? Is there two chains?
Distortion and well, most of the time. So they go to the bottom. You know what happens? They stop their motility,
Right.
so they become completely immobile for several hours, and then they become very misshapen. The best I can describe is they become. Have you seen like the morphed? I could show you some.
Are they still in chains or are they all unicellular?
No, they become unicellular and broken down.
Okay.
Some of them lice. You could see some of that.
Okay. Thanks.
Do they keep their flagella?
That's a good question. I have to ask Sylvan whether they keep their flagella. I'm not sure. I'm not sure whether I'm sure we couldn't tell that on the flow cytometer. And I'm not sure how much he observed microscopically to tell whether he, but that's a good question. I don't know. Tom.
I didn't get a chance to fill you in on some of the new things we know. For a long time, there's been a defect in the Japanese work on the involvement of actin, GDA with actin. When they did the experiments, they didn't examine whether there was any GDA left after they terminated.
Yeah.
Because risk of it being a transformation product of GDA.
Mm-hmm.
Now, this becomes very relevant. We now have been able to get characterization on an intermediate between GDA and acetic acid, compound we call goniodomic acid. And it's hard to work with because it's got a short half-life. However, if you consider the structure of it, the two things that are very similar are aflatoxin B1 epoxide, where it's got an intermediate that reacts with DNA in that case, where the intermediate half-life of it is maybe a second, maybe less, but it is the active substance in causing the carcinogenicity of aflatoxin. And so if you're just looking for things you can fish out and so forth, you would totally miss it. Another example is the epoxide leukotriene. Ben Samuelson got the Nobel Prize for figuring out how to characterize. I'm not sure we're going to get to that stage. But there again, the half-life of the compound, I suppose, was in milliseconds. But it was the heart of the biological activity of leukotrienes.
Okay, thank you. Any questions on that? Anybody?
Well, that concludes our session of Group 2, and we had a total of 20 speakers today. Thanks to everyone who came and gave talks, who talked remotely. Greatly appreciate you all, your time and attention today, and a huge debt of gratitude, of course, to our hosts here at VIMS, everybody that put effort into coordinating this meeting. Thanks to our behind the scenes hosts, Savannah and Lily. And of course, thank you, Kim, as always, for your gracious hosting here and for lunch. And thank you to everybody who joined us on Zoom. We will be posting a recording in the slides probably by next week sometime. we'll send an email out to let you know once they're all available. And of course, those of you sticking around for the ODU Planktoscope, we're going to go right down the hallway. So we'll see you soon. And thanks again for your time and attention. Have a great day.
Okay, and grab cookies and drinks on the way out if you'd like.
Let's go. I'm not gonna say.
Sure. Yeah, the plan is to get condos both in Norfolk and. Yeah. You've time to talk that it's 65. That's 65 degrees. Yeah. We're outside. Yeah.
What do you anticipate?
Still haven't heard yet. They verified.